A very good morning to one and all. It is indeed a happy occasion for us. On behalf of the Government of Maharashtra, Ismail Yusuf College of Arts, Science and Commerce, Jogeshwari, Mumbai, I, Mrs. Darakshan Sheikh, Head of the Department of Statistics and Organizing Secretary, extend a warm welcome to our distinguished guests, speakers, academicians, industry experts, participants and delegates, researchers, teachers and students to the inaugural ceremony of the two-day international e-conference titled Recent Innovations in Science and Technology, organized by all the science departments of our institution. Science as a field of knowledge knows no bounds. It is ever evolving, presenting new frontiers for limitless exploration. The avenues of inquiry are becoming extremely specialized and interdisciplinary. Technological innovations are happening at an exponential rate. Scientists are at the cusp of a number of amazing discoveries that could transform the world. Our conference is a humble effort in trying to have a peek into this emerging scenario. Through this conference, we wish to provide a platform for effective dissemination of knowledge, sharing of ideas, fruitful discourse, and intense, earnest, and unbiased discussions and deliberations for the benefit of all involved. We are extremely delighted and feel honored for this opportunity. In India, we have a tradition of invoking the blessings of Goddess Saraswati at the start of any auspicious event. I would request our Principal Madam Dr. Swati Vawal to light the traditional lamp for the formal inauguration of the conference.
तुषारहार धवला या शुभ्रवस्त्रावृता या वीणावर दण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्मविद्या ब्रह्माच्युत शंकर प्रभृतिभिः देवै सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा।
Thank you, madam. A constant source of inspiration and encouragement, our pillar of strength, the guiding light and driving force behind all our endeavors is our principal, Dr. Swati Varwal. I would like to request madam to deliver the inaugural address on this occasion.
Namaskar and a very good morning to all of you. Ismail Yusuf College is glad to welcome you all and to provide a platform through the International E-Conference, Recent Innovations in Science and Technology 2023 for the promotion and exchange of ideas towards innovations in scientific knowledge for a sustainable future. This conference aims to provide a platform and help the scientific community in understanding the contemporary development in the fields of analytical, material, pharma, data and environmental sciences. I extend my warm welcome to our chief guest of the conference, Dr. Sangeeta Ghumatkar Madam, Director, Directorate of Forensic Science Laboratories, Maharashtra State. I also welcome our keynote speaker for this conference, Dr. Arvind Deshmukh, former professor and head of the Department of Microbiology, Osmanabad Sub-Center, Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad. I am also very glad to welcome all the resource persons from India and abroad, participant researchers, academicians, students and the faculty at Ismail Yusuf College. I am extremely glad to mention that this International E-Conference, the response has been overwhelming and almost 550 plus participants have registered for it. The previous conference, E-Medfels 2020, hosted by our college, was also a grand success. Dear participants, I would like to share a few things about Government of Maharashtra's Ismail Yusuf College of Arts, Science and Commerce. The college is affiliated to the University of Mumbai and it was established in the year 1930 and is the fourth oldest college in the city of Mumbai. The college is situated on an hillock just opposite to Zogeshwari station and it has a huge campus of 54 acres. The campus is lush green with varied flora, especially the old and beautiful palm and banyan trees, a variety of bird species dwelling these trees, rippling brooks as well as reptiles. We have junior college, undergraduate and postgraduate courses in all the three faculties, that is arts, commerce and science. The college is also a center for Ph.D. in physics, zoology, chemistry, Urdu and Hindi. We run 14 self-financing courses and we have 6,000 plus students studying in all these various courses. Being a government college, the fee structure is very reasonable, attracting students from various stratas of the society. The college has an excellent infrastructure and the best of teaching and learning facilities. We are re-accredited by NAC with an A grade in the year 2017 with a CGPA score of 3.14. The college takes special care of socially and economically underprivileged students by holding special remedial coaching and by conducting various skill development courses. Ismail Yusuf College is also a center for Yashwantrao Chauhan Maharashtra Open University, wherein we run more than 14 UG and PG courses, under which 1400 plus students have registered. We all are aware that education plays an important role in the development of students and the progress of the nation lies in the aspiration of young minds. Besides empowering students with thorough knowledge of the subject, a focus is to be made on encouraging research interest in students. In order to pursue research in various fields, understanding of advanced instrumentation becomes the key factor. This conference aims to bridge the gap between theoretical knowledge and practical use of instruments, materials, data and technologies in designing scientific experiments. We at Ismail Yusuf College encourage interdisciplinary research and training for the students and faculty. The science departments of the college have taken up several research projects to obtain scientific knowledge. Our laboratories are well equipped with sophisticated instruments like HPLC, HPLC, LCMS, GCMS, AAS, CHNS automated analyzer, XRD, microwave parallel synthesizer, FTIR, NMR, and the list is long. Our endeavor is to nurture performance-based hands-on training on these instruments to the undergraduate and the postgraduate students. In this 2 days e-conference, eminent researchers and professionals from the industry shall be dealing with these various research aspects. The various sessions and deliberations in this conference by eminent speakers would certainly enrich and inspire young minds to find solutions to many challenges which they face during their research. I wish to compliment the organizers and all my science faculty members who have put in their ideas and efforts and have made this conference possible. I extend my best wishes to all the participants and I hope that you all shall greatly benefit from this conference. We have received 121 abstracts and I now announce that the abstract book for the conference is inaugurated. I wish you all a very happy new year 2023 and I pray that the conference RIST-2023 is a great success. Thank you one and all.
Thank you, Madam, for sharing your valuable thoughts on this occasion. I would now like to welcome and introduce the Honorable Chief Guest for today's inaugural event, Dr. Mrs. Sangeeta V. Ghumatkar, Director in Charge, Directorate of Forensic Science Laboratories, Mumbai. Dr. Ghumatkar holds a doctorate in biochemistry from Mumbai University, having expertise in the field of chemical and biological sciences. In a career spanning almost 3 decades, she has held numerous prestigious positions in the service of the Government of Maharashtra, such as Assistant Chemical Analyzer, Assistant Director, Deputy Director, Joint Director and Director. She has published more than 30 papers in national and international journals of repute and has also presented papers in international conferences in America and Germany. Dr. Ghumatkar has initiated new laboratory setups at the Regional Forensic Laboratory Kolhapur and Mini Forensic Science Laboratory Ratnagiri and has started new wildlife forensic DNA divisions at Nagpur and fast track DNA divisions at Mumbai, Pune and Nagpur. She has won various awards and garnered recognition for her contribution as Senior Woman Forensic Scientist by NFSU. We are indeed privileged to have Dr. Ghumatkar today in our midst as our chief guest. I would now request Dr. Ghumatkar to address the conference.
Good morning, everyone. I wish you a happy new year 2023. I am thankful to all honorables who have given me the opportunity for inaugurating this function of conference. I am delighted that my friend Swati Bawar has given me this opportunity. I thank from bottom of my heart all the honorables, all the scientists all over who are present here and abroad who are participating in this conference. This is an opportunity we give the students, scientists, graduates, postgraduates all over the country to interact on the platform or intellectual level. This is the platform where we exchange our thoughts. This is the world where we have to constantly read, watch and learn. Learn to the extent where we are going to innovate. We will be the innovating factors. Why is this a necessity of the today? Because in the world of this technology, as our conference name itself reflects, recent innovation in science and technology. Today is the need of this innovation in science and technology. Presently, I will tell you with my experience where I am working in this directorate for past 29 years, and I am as in charge director, where we come across n number of cases. We have learned in science, in the biology, for example, how blood grouping is done. But in the world of forensic, if you see that application of this knowledge, what you have acquired, will have to turn into wisdom. How? When a crime scene you are visiting, you are not only have to have what is the blood group on the weapon, the clothes. Here the challenges start. Here your knowledge will be used. Here your wisdom will be used. The accused try to wash their clothes, wash the weapon, and you have to find out from this washed clothes the blood group and link the cases. In the forensic, we see many cases even in ballistic, whether this bullet has come from this firing arms. I have attended 26/11 bomb blast, German bakery bomb blast, where all chemistry comes into picture. Which RDX molecules were found have to be connected to the crime scene, and also you have to pose and impose and give your reports in the court of law. So your science knowledge is going to be very important here, and your wisdom, how you will apply, how your research implication you will be making in this world of science, which will be helping you further in your investigation. So every student who is watching, participating in this technological and innovative science conference will be surely benefited, because this will be impacted on their mind, and in turn which will they imply and implicate in their life, to their work, in their further research, and wherever to bring the humanities into good surrounding. Make this planet the best place to stay. So I announce that I inaugurate and start this conference, which will be a great success and bring a productive conference where everyone will be benefited from this conference. I wish all the delegates, all the honorables, that they enjoy and take home a really good wisdom and knowledge full technological and scientific research oriented conference memories. Thank you all.
Thank you, madam. It was a pleasure to hear you speak. I'm sure that your talk will inspire a lot of young students and researchers to develop interest in the promising field of forensic science. We also thank you for your good wishes for the success of the conference. It is an honor for us to have the keynote address of this conference delivered by an extremely eminent personality from the field of education, Dr. Arvind Madhavrao Deshmukh. Dr. Deshmukh is currently President of the Microbiology Society of India and is also retired Professor and Head, Department of Microbiology, Dr. Babasaheb Ambedkar, Marathwada University, Sub-Center, Osmanabad. Dr. Deshmukh's teaching career has spanned 37 years. His research experience in areas such as nanotechnology, biogas and dye degradation, to mention a few, is indeed unparalleled. He has 82 paper publications against his name, 32 of them being in international journals, and has completed 1 major and 2 minor projects, written 4 and edited 7 books, and has had 17 Ph.D. awardees under his guideship. He has organized 76 state, national and international level conferences, seminars and workshops in India and abroad. He has been resource person in 354 conferences and delivered talks on radio about biofertilizers, biopesticides and environmental pollution, among other things. His articles have also appeared in numerous Marathi language newspapers. He has visited many foreign countries for presenting papers and as invited speaker. He has been involved in arranging virtual lectures of eminent scientists from all over the globe and was also very proactive during the pandemic by making many informative videos on COVID-19. It is surely our good fortune to have Dr. Arvind Deshmukh deliver the keynote address of our conference. With this, I invite Dr. Deshmukh to kindly take over for the address.
Thank you. Thank you very much. Thank you, madam, for introducing me. And at the outset, I would like to appreciate your college for arranging the excellent e-conference where I came to know you have got many abstracts, and in the conference many eminent speakers are going to talk, and really the title
Good morning, sir. See, you're not audible right now. Could you please check your connection?
Sorry for the problem, technical problem. I'm extremely sorry. Now I think that you can listen me. So again, I have joined and I can so please share. Yes, next slide, please. Next slide. Sorry for that. Yes. So global problems. Again, I'm coming to the biotechnology. Now, the world is facing problem. Which problems world is facing? Food problem, environmental pollution problem, energy problem, and health problem. Understand now, the food problem is the major problem in the world and specifically to India. Again, I'm coming recent innovations in biotechnology. How we can solve the food problems throughout the world? We can solve the food problem only and only with the help of biotechnology. No other technology will help you. Same thing is about environmental pollution. I am discussing in detail. So I will go in the next slide. So energy problem, health problem, everywhere biotechnology and innovations in biotechnology will help you. Next slide, please. Correct. So what I'm telling, now what are the innovations? If you ask the innovations, recent innovation, prefix bio word to any technology. Understand? Prefix bio word to any technology and it is a biotechnology. And it is a innovations. You take a fertilizer, it is a biofertilizer. You take a leaching, it is a bio-leaching. Pesticides, biopesticides. Steel, bio-steel. Remediation, bioremediation. Cement, bio-cement. Plastic, bioplastic. Fuel, biofuel. Informatics, bioinformatics. Instrumentation, bioinstrumentation. Sensor, biosensor. Batteries, biobatteries. Energy, bioenergy. And likewise, many I can give actually more than 100 words where there are innovations in the technology. And it is, I'm going to talk, but you see, 45 to 50 minutes is not enough to explain everything, but I will go shortly. I will touch to the every topic. And later on, you can go on Google and you can search details about every aspect of the innovations in biotechnology. Let us go to the next. Correct. First, I will come to the antibiotics. Why? You see, whole world was facing the problem of corona, you know that. And corona, again I will come to corona. Where from corona virus entered on the earth? Probably Wuhan Institute of Immunology. And what work they were doing in the Wuhan Institute of Immunology? In the Wuhan Institute of Immunology, already research on coronavirus was going on. And again, I'm telling you, you must know the innovations in biotechnology. The research, genetic modification research is banned in America, but it is not banned in China. And what is about India? It is not banned in India. The reason is that India is not capable to do the genetic research on human being or advanced research on microorganism. But in China, they can do it. And what is the innovations we can say? Going ahead the nature. Understand? On the coronavirus, scientists were doing the work going ahead the nature. Meaning is that they were modifying the coronavirus, and they were going ahead the nature. Now you will say, why they are going ahead the nature? Because they want to find out the remedy, treatment, medicine, if the coronavirus mutated in the future. That's why we have to do the work going ahead the nature. And probably organism from Wuhan Institute of Immunology escape and cause problem throughout the world. Now you see why I'm coming to the innovations in biotechnology. Because now many scientists are telling next pandemic will be antibiotic resistance organism pandemic. Mind it well. After 4 or 5 years, antibiotic resistant organisms are dominating throughout the world. There will be many deaths throughout the world. And how we can stop the deaths? The antibiotics will not be working now. Streptomycin, penicillin already is now not in work because most of the organisms are resistant to penicillin. Now, new antibiotics are discovered by the scientists, but I'm going to the last point of this slide. Bacteriophage theory, bacteriophage therapy is the innovation now. Meaning is that you know the bacteriophages. What are the bacteriophages? Bacteriophages are killers of bacteria. Now, very simple technique here. Enemy's enemy is our friend. What do you mean by it? It means that who is our enemy? Pathogenic bacteria is our enemy. We want to kill pathogenic bacteria. And if we want to kill pathogenic bacteria, we will use bacteriophages to kill pathogenic bacteria. And that is called as a bacteriophage theory. And I will tell you, research is going on in India and abroad, and there is a success now. So in the future, after 10 years, antibiotics will not be given to you and me if you are suffering. The suspension of bacteriophage will be injected in your body and in my body. And bacteriophage will attack on the bacteria, pathogenic bacteria, and will get rid from the diseases. That is called as innovations in antibiotic therapy. Next slide, please. Bioterrorism. I can talk hours together on bioterrorism. Whenever I'm talking about the innovations, I will talk also about the awareness about the innovation. In India, neither people are bothered, people are aware, not people are worried about the bioterrorism, and not our politicians, not our administrators, they are worried about the bioterrorism. As a president of Microbiologist Society, I have written letter to Honorable Prime Minister. Please frame the rule regarding bioterrorism. Why? Because USA have framed the rule, but India has not framed the rule up to the time. So what is a bioterrorism? It is again innovation. Innovation meaning is that in next era, next years, coming years, the war, weapons in the war will be bioweapons. Now it will not be atomic bomb. It will not be chemicals. The weapons will be biological weapons will be dominating. And whether they are used, yes, they are already used by many countries. And some scientists were telling that corona is also the biological weapon used by China. But as a microbiologist, we don't believe such fake news. It was not used by China as a bioweapon. Probability is there. It might have escaped from one institute of immunology and cause a terrorism in the world. So, bioterrorism, I will talk on the bioterrorism. What is bioterrorism? Use of pathogenic bacteria, viruses, fungi, protozoa, or toxins produced from it. If used for terrorism, we call it as a bioterrorism. What are the advantages of bioterrorism? The advantages is that Biological weapon kill only target. It will not touch to the animal. It will not touch to the plant. It will not touch to the building. It will not touch to the infrastructure. Meaning is that when bioweapon is used, will kill only human being, and ready infrastructure with animals, with plants, it will be in our hand whenever we'll conquer the country. That is the advantage of bioterrorism. Second advantage of bioterrorism, if we use 1 kg of bioweapon, within 1 month, it will multiply, and it will become 100 kg of bioweapon. Meaning is that a very small amount, if added in the environment, it will multiply itself. So, small amount of bioweapon is required to attack on another country. Third advantage is that whenever you use bioweapon, it will start its effect after 7 days, after 8 days. And during that time, the bioterrorists, they get time to escape from that country. Means they have used a plague organism, they have used Vibrio cholerae organism or Bacillus anthracis organism. It will take some time to multiply in the environment and to cause its effect in the environment. During these 7 days, during these 8 days, bioterrorists can escape easily from the country. That is the advantage for the bioterrorism. And that's why in the future now, India should look after the bioterrorism, to avoid the bioterrorism. And I will tell again, I will request to this your college, please arrange awareness regarding the bioterrorism, because awareness on the bioterrorism is badly needed in India. The Bacillus anthracis organism have been used in USA as a bioterrorism weapon. In 2006, in 2003, already there were many attacks on very important personality in US by Bacillus anthracis organism. And that's why USA made a rule, Parliament has made a rule to avoid the attack by the bioterrorism. But unfortunately, India is not prepared. Again, I will remember one sentence. Whenever there was a corona on 20th March 2019, our Prime Minister addressed India, and in first lecture, he told that India is at all not prepared to face the corona disease. Same situation will be there. India is not prepared to face the bioterrorism, and bioterrorism is an innovative way to attack on the human being. Next slide, please. So one by one, I will go. Now you see. Biofertilizer, you will say, sir, what is the innovations in biofertilizer? We know biofertilizer. We know azorazo. We know acetobacter. We know phosphate solubilizing microorganism, or blue-green algae, or VAM, meaning is that vesicular arbuscular mycorrhiza. They are already in market. So what are the recent innovations in that? I'm going to tell you recent innovations. In old time, biofertilizers were solid biofertilizers. In the middle picture, center picture, I'm showing the solid biofertilizer, like a vermicompost, like the fertilizers mixed in humus, mixed in lignite. Lignite was black in color. It was packed in polythene bags, and farmers were used. The disadvantages of the old biofertilizer was hands were becoming black, and farmers were not, they were reluctant to use this solid biofertilizer. Now, what are the innovations? Now, liquid biofertilizer, first picture, bottles I have shown. Liquid biofertilizer is available in the market. Now, the era of solid biofertilizer has gone. Now, most industries in India also, in Maharashtra, producing liquid biofertilizer. Again, more innovations. Innovations are that the biofertilizers are produced in capsule. Again, I will tell Vidya Pratishthan Baramati is producing biofertilizer capsules. In Pune, biofertilizer sticks are available, biofertilizer capsules are available, and you can decide the dose. So per pot, one capsule can be used. So it is easy to use in the city area, in the urban area. That's why these are the innovations of biofertilizer. Some scientists are trying to produce a biofertilizer which is resistant to the temperature, which is resistant to the dryness, so that it will work in the dry soil, it will work during the summer. Such type of biofertilizer will come in market. That is called as a recent innovations. Again, I would like to tell you few things, because you see, what are the innovations? Today's topic is innovations. Here, biofertilizer word is a old word. Now, microbial inoculant is a new word to it. Biofertilizer is an old technology. Now, you should not also use biofertilizer because why? Why microbial inoculant is a new technology? Because microorganisms are not fertilizers. Microorganisms are producing, releasing nitrogen, phosphate, or sodium, or whatsoever required, potassium. They are releasing. They are not acting themselves as a fertilizer. So like chemical fertilizer, we should not use word biofertilizer. We will use word microbial inoculant for the production of fertilizer. So that is a biofertilizer innovations. Next again. Next slide is biopesticides. You see. Again, biopesticide is the same. Enemy's enemy is our friend. Understand? Again, chemical pesticide. If you remember Amir Khan, because every one of us know Amir Khan. Amir Khan is a very good actor, but social worker. Before 10 years back, he organized one event on TV, Satyameva Jayate. In Satyameva Jayate, one episode was dedicated to health hazards of chemical pesticides. So, chemical pesticides are very hazardous to the health, causing cancer. And government is telling, scientists are telling, social workers are telling, don't use chemical pesticides. In all developed countries like USA, like UK, like European countries, they are not using chemical pesticides. You see, they are not using chemical pesticides. They are using only biopesticides. Again, I will tell you. You will ask, what are the biopesticides? Biopesticides, BT. BT means Bacillus thuringiensis. Again, I will come to the innovations. If you know Bacillus thuringiensis, what are the innovations I would like to tell you? But trichogramma. Trichogramma is an insect, and eggs of these insects are available in the market. They are sprinkled on the card, and that card is called as a tricho card. Tricho card is available in the market, particularly from Bangalore. Biopesticide Institute is there. Card is available. So, trichogramma is a biopesticide. Trichoderma, you see, trichoderma is a miracle fungi. And people are earning lakhs of rupees by producing trichoderma. Trichoderma is a fungus protecting the plant from pathogenic microorganisms, as well as it is producing phosphates. It is solubilizing phosphates, so phosphate solubilizing microorganism. Then, nuclear polyhedrosis virus, NPV. NPV is also produced in Sangli. There is an industry, and NPV production is a very simple. and it is eco-friendly, because you see, whole world is developing nowadays. But now one word is important, eco-friendly. Meaning is that whether the tools we are using, they are eco-friendly tools, NPV is eco-friendly. So the eco-friendly NPV should be used. Now, what are the innovations I will tell you? The innovations are that Bacillus thuringiensis have a gene for the production of toxic protein, that is cry protein. It is called as a cry gene, cry protein. And this gene is transferred from Bacillus thuringiensis to, it is transferred to the plants. And it is already transferred in cotton, that is called as a BT cotton. It is transferred in brinjal, it is called as a BT brinjal. It is transferred in apple, called as a BT apple. Now, these are genetically modified plants, now cultivated by many countries. Unfortunately, in India, only the BT cotton is allowed. BT brinjal is not allowed in India. It is allowed in Bangladesh. It is allowed in many countries, but not in India. Why? Because you and I myself is responsible for it. We failed in creating awareness. In India, traditional peoples are more. They are opposing genetically modified food, telling there are many disadvantages of the genetically modified food. If disadvantages are there, from many years, they are consumed in all these countries. Nothing happened to these countries. Why you are telling unnecessarily that BT genetically modified foods will cause a problem to the human being? So it is wrong. Now we will convince the government, and I have written a letter also to the government, please allow. And there are many companies in India. One example I will tell you, Barwale company from Jalna, they are waiting for the genetically modified brinjal, but India is not giving permission. So genetically modified food is innovations. And one more innovations I will tell you, one more, that is a red colored rice. You see, vitamin A rich rice. Vitamin A rich rice is produced in Philippines, but it is not allowed in India. Why it is not allowed in India? Again, genetically modified. Actually, vitamin A rich rice, called as a golden rice, named as a golden rice, it will remove the nutritional deficiency. It will fulfill the vitamin A requirement, and poor farm people in India will not suffer from the deficiency of vitamin A. But it is not used in India. One more again. Next slide, please. Next slide. Bioplastic is one more thing again. You see, we are using bioplastics. Sorry, we are using chemical plastics. We know that chemical plastics are causing problems, causing nuisance, particularly at a tourist place. I was in one environmental committee of Mahabaleshwar. At that time, the problem was raised by the ministers of Maharashtra that in Mahabaleshwar, plastic pollution is there. How to remove the plastic pollution? And it is not the problem only in tourist place, on rail platform, on ST stand, and where the people, crowded peoples are there. Plastic pollution is creating a problem. And for that purpose, we should go for the bioplastic. So the bioplastic, in Maharashtra, there are 50 industries producing bioplastic. You go in the shop, you will get a plastic, the bioplastic bags, organic plastic, biodegradable plastic. So bioplastics are biodegradable and most important, eco-friendly. Polylactic acid, polybeta hydroxyl butyric acids are used in preparation of bioplastic. So the bioplastic should be used. Again, I am requesting today to you, whenever we are discussing the innovations, recent innovations in science and technology, the bioplastic plays a major role. And all laymen should be aware about the hazards of chemical plastic. I will not talk more on because limited time is there. But chemical plastics are causing many hazards to the human health, animal health, fishes health, and aquatic life health. It is attacking badly. So we must use bioplastic, and they are available in the market. From today, I will request to all listeners, go in the market and purchase plastic bags which are biodegradable. Next slide, please. Probiotics. Again, you see, I'm telling importance of the probiotics. Now the probiotic industries are there. Yesterday only there was the show on the TV regarding the probiotic producing industries, and probiotics are getting huge market nowadays because people are realizing the importance of probiotic. I will request all listeners, you try to avoid to consume the thumbs up, Coca-Cola or other soft drinks. They are injurious. Go on the Google, search theory of longevity. Meaning is that if you want to live longer life, you must consume probiotics every day in your life. And probiotics makes you healthy. Probiotics keeps you young, and probiotics maintain your health. So particularly old people, aged people should consume probiotics. And there are many industries of probiotic. Even in Maharashtra, there is an industry in Chikrur. Lactobacillus, leuconostoc, and sporolactobacillus. So these industries are the industries present in Maharashtra, India. These industries are present in India and selling their products in the market with the different commercial names. There are many advantages of the probiotics. The time do not permit me to tell you the advantages of the probiotic, but sporolactobacillus is an innovation now, because it is a endospore producing lactobacillus, and it is now used as a tool by many industries. Next now, correct? Single cell protein. I will tell you the very interesting thing about the single cell protein. And if you know the importance of single cell protein, in today's my lecture, I will say that my lecture is at positive side. Single cell protein, why it is required? Protein deficiency in diet can be supplemented by using microorganisms, and spirulina capsules are available in the market. Again, I will request all listeners, go on the Google and search spirulina. You will get hundreds of industries in India producing spirulina. I purchased one pocket, and in that one capsule cost 20 rupees. One capsule cost 20 rupees. You will say, sir, why to purchase this costly medicine? It is not a medicine, it is a food. And instead of eating a dinner, you take one capsule of spirulina. It will fulfill all your requirement whenever you will get by eating one dinner. Because spirulina contains all amino acids. Spirulina contains all vitamins. It is a palatable. It gives a protein, highest protein, more than the soybean, more than the egg white. It is the highest protein content. So spirulina capsules and powders are available in the market. And if you are suffering, for example, you are suffering from a typhoid, and doctor will give medicines and you are now covered with a typhoid. You are recovered from typhoid. Now you are feeling weakness. Whenever you will go to the doctor, doctor prescribes you spirulina tablets. Meaning is that you take spirulina whenever you are having weakness. Whenever you are having normal health, you are not consuming spirulina. When I was a child, at that time, we were eating fruits only when and when I was feeling a weakness, not as a routine diet. Like
Sorry for the delay again. Sorry for the problem. Can you listen me?
Yes,
you
sir, you're
listen?
audible.
Okay, thank you. Sorry. So after single cell protein, next slide, please. Next slide. Bioleaching. You see, bioleaching is a very, very innovative. The reason is that nowadays there is a major problem of e-waste. You take a next slide, please. Next slide. E-waste. Electronic waste is a world problem nowadays. You see, because before 20 years, no one was having mobile. Very few people were having only computers. In 1986, there was only, in 1996, there was only one computer in my college. And I have to take a permission to use the computer. I have to write application to the principal. Principal allowing me to use the computer only half an hour or 1 hour. That was the situation before 25 years. What is the situation nowadays? Nowadays, we have plenty of computers, plenty of laptops, plenty of mobiles, plenty of hard disks and pen drives. Now, what is the problem? I will tell you the problem. The problem is that this electronic waste we are throwing on the garbage. And whenever we are throwing on the garbage, it will get degraded there with the microorganism. Release metal. What are the metals in the electronic waste? The iron, lead, and aluminum, gold, silver, and many other metals. These metals will enter into the environment and ultimately they enter into water bodies. In the river water, in the well water, in the lake water. And whenever we consume that water, ultimately it will enter in your body and my body. And it will cause a bioaccumulation. And they are toxic. They cause cancer. They affect on liver. They affect on kidney and many other organs of the body. So the e-waste should not be allowed to enter. In 2009, Central Pollution Control Board have made a law. Which law? Regarding the disposal of electronic waste. Now, what I will suggest you, that is, or what you many listeners among them, I will inspire you to work on the bio-leaching. One of my student, Ph.D. student, already worked on bio-leaching of electronic waste. And he got successful results. Many bacteria, particularly chemolithotrophic bacteria, can dissolve, release the metals, and we can create wealth from the waste. Mind it well. Means if the gold is there, we will recover the gold from the electronic waste. And gold has an economical value. Silver has an economical value. So the electronic waste treatment by microorganism called as a bio-leaching is an innovative approach for the treatment of the one type of waste. Now, next slide, please. Genetically modified food, I have told you. Just I have discussed it. And I will, as I have told you, the golden rice is not allowed in India, and genetically modified foods not allowed in India. And what a joke. Again, I will tell you one joke I will tell you in biotechnology. Government is telling, BT cotton we have allowed because BT cotton is not allowed as a food. It is not consumed as a food because cotton is not a food. So it is telling that the BT cotton we have allowed in India because it is not consumed as a food. But understand, the leaves of BT cotton are consumed by the animals. And whether there is an adverse health hazard on the animals from last 20 years, no. Second, the cotton oil is consumed by some parts of India. Means cotton oil is a food. It is consumed, which is produced from BT cotton. It is not causing any hazard. But government is telling that we will not allow the genetically modified foods in India. We have allowed BT cotton because BT cotton is not a food. So these are the jokes, and we biotechnologists must fight to the government, must write to the government. We must create an awareness in the society. And today's seminar is a one attempt to create an awareness regarding innovations in biotechnology and science and technology. Next slide, please. You see, the many slides are there. I will try to cover within 10 minutes now. BT cotton, already I explained. Next slide, please. Biocement. You see, one of my students, she is working on biocement. She completed her PhD from Bath University. Her name is Jamal Purey. You go on the YouTube and search regarding biocement. Biocement is a very innovative approach. Whenever there are cracks in the building, instead of using cement to seal the crack, you use biological culture, microbial culture. This microbial culture will fill up the gap, and it is having more strength as compared with the normal cement. So biocement is a very, very innovative approach. Whenever your title is innovations, recent innovations, yes, I will tell biocement is a recent innovation. And whether it is used, yes, biocement is already used by European countries. but not in India. But it should be cheap, little, it is costly. Now, scientists are trying to make it cheap, and it will be also common in India. Within 10 years, it will come in the market, and they will tell you, if your building is having crack, instead of a normal cement, use biocement, because these are the microbial activity producing cementing substances, and these substances seal the crack. All detail regarding the mechanism is given on the YouTube. You can go on the YouTube, you can go on the Google, search it, and you will get the mechanism, you will get the various bacillus species which are used. Urease activity is mainly responsible for biocement. Next slide, please. Biofuel. Understand? You will ask me, what is the innovation, sir? Yes, I will tell you the innovations in biofuel is that our honorable Transport Minister of India, Nitin Ji Gadkari, regularly asking and telling that I want biofuel. He is appealing to the biotechnologists of India that please produce biofuel, because he has given a permission to use 15% ethanol in your vehicle. So the biofuel and microbial fuel cell, MFC is not in the market. Scientists are working on MFC. Many students are working on MFC at present. So biofuel and MFC are the innovations. Means Nitin Ji Gadkari has given permission to use 15% ethanol in the fuel of the vehicle. But he is telling that I'm not getting enough amount of ethanol in India. So it is a duty of you and me, as of all biotechnologists, to produce a bioethanol and to start our vehicle by using bioethanol. In Brazil, the vehicles 100% they are using bioethanol. If they are used in Brazil, why India should not be capable of producing bioethanol as a biofuel? Then next slide. MFC, as I told you, MFC is at a research level. Gut microbiology, very innovative and very interesting, you must know. And maximum research is going on in India on gut microbiology. Because the gut microbes, what is the amount of gut microbes? Gut microbes, 150-fold of our genome means that DNA. Because if we estimate the DNA of our body DNA is less and microbial DNA is more. Microorganisms on our skin, microorganism in our nose, in the mouth, in the intestine, in the rectum, there are plenty. And maximum research in India is going on gut microbiology. You will ask me, what is innovation, sir? I was in Nagpur. And from SIMS, one scientist told me, Rajpal Kashyap, he is a scientist there in SIMS. He told me, sir, now we are doing research on fecal capsules. What do you mean by fecal capsule? Fecal capsules meaning is that from feces of human being, microorganisms are isolated. And from that microorganisms, capsules are produced. Meaning is that from gut microflora, capsules are produced. Now why? It is known that if the person's nature is good, for example, the APJ Abdul Kalam, his intestinal flora might have the excellent microorganism. Whenever the person is talent, whenever the person is a leader, whenever the person is a very good, at that time, his intestine also bears good microorganism. So from the fecal matter of such person, the microorganisms are isolated, and these microorganisms are converted into capsule, and that capsules the normal person should take, or a criminal person should take, his behavior will change. Because these microorganisms present in our gut, they are producing neurotransmitter. So these many hormones, many hormones are there. They are produced by microorganism, and they repress the pathogenic organism also. That's why it is said that if you are suffering from any disease and if you are recovered, you should consume curd, leuconostoc, lactobacillus to establish the normal flora on our body. So normal flora in our body is re-established, and to re-establish, fecal capsules are highly useful. So gut microbiology is a very, very important, as again there are time limitations. I can deliver a talk, 1 hour talk only on gut microorganism. And many scientists in India are working on gut microbiology. Let us go to the second, because time, freedom from AIDS. You see, recent innovations is that stem cell technology. These stem cells, if introduced in the persons which are suffering from AIDS, because certain human beings are there, they produce resistant WBC. Stem cell from certain human beings are taken and they are introduced in the patient. If they are introduced in patient, it is observed that AIDS patients get recovered by transplanting such stem cell. So it is the best medicine. Again, I will tell you, now the many innovative approaches are there to treat the patient. Stem cell technology is one of the innovative approach. Next slide, please. Biofarming. Now it is not a FARM, it is a PHRM. What is a biofarming? Biofarming meaning is that pharmaceutical changes in the food material. Next slide is also on that the same topic. It is a edible vaccine. Edible vaccine meaning is that the vaccine now, you know that the insulin hormone, in old time, insulin hormone was extracted from the animal pancreas. Now, the insulin is obtained from microorganism, genetically modified microorganism. Likewise, it is possible in the future to produce edible vaccines, and then small children will not be worried, because now vaccine is injected and the needle are used. In the future, no needle, no injection, the vaccine will be present, this gene will be present in the banana, and for vaccination, banana will be given to the children. That is a time will come very soon. Research is going on. Next slide, please. It's about 51 minutes are there. I think I should conclude now, because I have delivered a lecture about 50 minutes. Within 5 minutes, I will cover bioluminescence. Certain microorganisms are having luciferase enzyme, and as a result of it, they are bioluminescent organisms. Scientists are trying to transplant that gene into plant. Is it successful? Yes. In Lucknow, the institute has transferred this gene into tobacco plant. And now tobacco plant is glistening and producing a light, emitting a light. So such plants can give light in the night. Sad. These innovations are possible in the future. Such a luciferase producing gene will be transferred from such microorganisms. It will be transferred into plant. Transfer of the gene is very simple, but expression of gene is difficult. Scientists are working on it. Scientists got a success on that. And now the plants will be emitting the light in the future. And the whether is it possible for the human being, whether such gene can be transferred in the human being? Yes, it is possible, but there is a ban on such research. Any government throughout the world do not allow genetic engineering work on human being. So it is not possible. One scientist in China tried before 5 years, then he was in prison. So no chance of work on human being in genetic engineering. So bioluminescence transfer of gene in plant is possible in the future. Next slide, please. Bioremediation. I will tell you, we are facing a problem of pollution, solid pollution, liquid pollution, organic, inorganic, and there is a solution to it. If you remember Anand Chakravarti was an Indian scientist. He was from Bengal and he was a resident in USA. He died before 3 years. During Corona period, he died. So Anand Chakravarti, he was the first scientist throughout the world who produced a genetically modified microorganism called as a superbug, and he applied for the patent. Then patent office in USA denied. Why? They told that organisms and bacteria, you cannot patent. But he argued that this bacteria is a genetically modified bacteria. You should give me patent. The case was filed in the court, in the Supreme Court of USA, and Supreme Court told that, yes, you must give patent to Anand Chakravarti. He is the first person in the world to get patent of microorganism. And this superbug means, you see in the picture I have shown 2 workers. They are sprinkling this superbug on the surface of, near the banks or seashore, near the seashore. There is a contamination of the oil. This blue colored oil on the water surface. It is very difficult to remove this pollution, but microorganism can remove the pollution. Superbug can work dramatically and interestingly. Next slide. Because bio-innovations, bio-bleaching means removal of color, work is going on. Next slide, please. Food microbiology, you see many things are there in the food microbiology, particularly in food fermentations. New foods are coming in the market, and in India, huge market is there on the food microbiology, because in India, 140 eating mouths are there. That's why huge market is in India as compared to any other country. And many food products are coming in market with a good aroma, good smell, good taste. So there is a tremendous scope in food microbiology. So food with a medicine value. Next slide, please. Genetically, it's the same thing. I will go next slide, please. Because tissue culture of the plant. As I am not a botanist, I will mention it. I will not go in detail, but tissue culture of plant have a huge market in India. And the people are earning crores of rupees by tissue culture of plant. Next slide. Biosensor is an innovative approach for used in analytical instruments. And Trapti Gokhale from the Dubai, she is working on a biosensor. So many scientists are working on a biosensor, innovative approach for in the analytical instrument. Next. Bio business. You see, now instead of a normal business, bio business will increase. And I will request all listeners, encourage your student to take a degree of bio business. Bio business now has increased tremendous, not in India, also in the world. Next slide. Last, I think. Vaccines, I will not go in vaccines, please, because the time do not permit. Bioethics only. Now, ethics values also change. Whenever I am telling innovations, you see, ethics are changing time to time. In old time, people, the ladies were wearing saris only and ghungat. Nowadays, they are wearing Punjabi. meaning is that ethics are changing day by day. Likewise, bioethics are there. Bioethics meaning is that the question was raised in books also it is given. If the banana is there and have genetically modified banana, gene transferred from animal, if the gene is transferred from animal in banana, now banana is a vegetarian or non-vegetarian, ethical value. Are you getting it now? Whether banana should be called as a vegetarian or a non-vegetarian, because banana is a vegetarian, but the gene is transferred from animal, it has become a non-vegetarian. Many ethical values are there. There are books on the bioethics. You go on internet search, you will get many examples of bioethics. And that's all I think. Last slide, please. Yes. So thank you very much for listening me for 1 hour. And although there was, I'm very sorry for the technical disturbance was there 2 times, but you are beard with my mistakes. Sorry for it. Thank you. Thank you. And again, I thank Swati Wavar ma'am particularly. Really, she is a dynamic lady and she is always encouraging such activity. And Sheikh ma'am was also continuously in touch with me. Thanks all. Thank you, everyone. Thank you, all listeners. Thank you. If any question is there, I can answer.
In his address, Dr. Deshmukh has touched upon multiple avenues of inquiries in the field of biotechnology and how the many wonderful innovations in this field are having profound impact on the ever-evolving global scene. His address was interesting as well as informative, and as such has set the tone for fruitful discourse at the conference. Thanks a lot, sir. We are much obliged. We will now stand to attention for the national anthem of India. Thank you all. This brings us to the conclusion of the inaugural session of the conference. Without any delay, we will directly proceed to the first session of the first day of the conference. Thank you all. This brings us to the conclusion of the inaugural session of the conference. Good morning once again. May I have the pleasure of welcoming in our midst our first speaker of the day, Dr. Dharmesh Reykundaliya, with a brief introduction. Dr. Reykundaliya is an academician par excellence. His educational qualifications and innumerable noteworthy achievements are such that no measure of allotted time during the course of this conference can really suffice for a justifiable account of all of them. Nevertheless, a mention of some of the most outstanding ones is absolutely essential. Dr. Dharmesh Reykundaliya is Assistant Professor, Department of Statistics, Sardar Patel University, Vallabh, Vidyanagar, Gujarat. He has a doctoral degree in reliability-oriented designs. He has research experience in other fields such as genetics and statistical modeling as well. He has expertise in more than 20 diverse fields of statistics, such as statistical quality control, business statistics and marketing research, operations research, biostatistics and pharmaceutical science and biosciences, designs of experiments, econometrics are just a few which I can mention now. Dr. Reykundaliya has published 33 research articles in numerous journals, written 2 ebooks, 7 consultancy project reports, has 7 research projects from UGC, the government of Gujarat, and the university, and has 9 awards against his name. He is also currently guiding 1 student for Ph.D. Apart from attending and presenting papers in numerous conferences, seminars, and workshops, he was an invited guest speaker in 165 of them. He has also been organizing secretary in 21 of these events. He's also a life member of Gujarat Statistical Association and 3 more associations of the like. Dr. Dharmesh Reykundaliya is a well-rounded personality, devoting himself wholly to academics and to student-centric activities on and off the campus with eager enthusiasm. With these words, I invite Dr. Reykundaliya to engage this session. Thank you. Dr. Reykundaliya.
Thank you, madam. Am I audible?
Yes, sir, you're audible.
Yeah. Thank you very much for a nice and kind introduction. And I would like to thank my sincere thanks to the organizer for inviting me over here to deliver a talk on a biostatistics, which is in the recent innovation in the science and technology, Ismail Yusuf College of Arts, Science and Commerce, Mumbai. Well, the title of the topic is biostatistics, but I had little bit, of course, when I'm talking about the biostatistics is nothing but application of statistics in the field of biology. But a few days back, I came across with one of the person who is working as a professor in one college. And he said that, sir, we are from the non-science background, or we are from the non-statistic background. And we are facing a lot of difficulties that which statistical technique should be suitable under which condition. And that is happen to everywhere, whether it is from bioscience, pharmaceutical science, finance, or market research, or any field, if you are talking about that. So what I will do, I'll try to restrict myself the use of the application of statistics in the field of various fields, especially trying to focus on the biostatistics. Yeah, next, please. So having heard from many non-statisticians, while analyzing the data, they are facing a lot of difficulties due to lack of knowledge in the field of statistics. Maybe there are no such books available which can guide and cover all possible techniques for the data analysis with proper logic, theoretical, as well as computational through different softwares. So there are many books of statistics available in literature, but maybe that books are discussing about the theoretical statistics. Some of the books are available, they are purely talking about the software, but there are very few, or you can say that there are very less book, which is a blend of both the giving the idea about the theoretical statistics as well as their application using the softwares and how to interpret that. So in this presentation, I'll try to present a brief survey. Yeah, please go back. In this presentation, I'll try to present brief survey on such statistical modeling technique with respect to nature of data. Most of the time, what happened, I have come across to the people who are not having a background of statistics because of the several softwares are available like SPSS, Minitab, R, SAS. So what they do, they just supply the data, they click the particular output or the select the technique and click it, and they will come across with the output. And they are saying that, okay, sir, this is the lot of analysis which we have done. Now the issue is that which analysis is helpful to us. So now the question is that, okay, whenever you have a data, first you have to understand the domain of the data. Suppose if I'm dealing with the biostatistics, if I'm dealing with the bioscience, I have to understand the nature of the data. If I will not understand the nature of data, how the data are generated, how the data are collected, I am not in that position to apply the suitable technique. So softwares are there. Softwares can minimize your effort of computation, but it does not help you or it does not guide you that which statistical technique should be used at what time. So I'll try to see that according to the nature of data, which statistical technique should be used. Also try to explain appropriate method which is applicable under the different situation. So this presentation is classified with respect to the measurement, level of response and explanatory variables and corresponding technique possibly by demonstrating with R language. So I'm not going to discuss R, but I had the analysis of R and that I will help you that how you can interpret this particular output. Next, please. In this presentation, I'll cover the following topics. We'll talk about the scale of measurement. Then we'll talk about the choice of statistical analysis. And because in statistics, we always deal with whether the data is random or not. So to independentness or the randomness of the data can be assessed based on the run test. We'll talk about that. Then we usually say that the response is coming from a normal distribution. So if I wish to see whether the data is coming from a normal distribution or not, we can have a goodness of fit test. And that test is nothing but the one sample Kolmogorov-Mindoro test. And of course, we have a test of hypothesis, which is classified into the parametric as well as the non-parametric. So I will discuss the difference between both of them and specifically focus about the parametric test, one sample T test. And if time permit, we'll discuss more. Next, please. So statistics has its own vocabulary. Many times what happens when I discuss with the people, they said that, sir, we are the person belong to the non-statistic background. So we don't know because we don't like mathematics. That's why we opt for the biology. But I would like to convey that the statistics is in your day-to-day life. You cannot say that I don't know the statistics. In every day-to-day life, we are using the concept of statistics, but we don't know that this actually concept and this point is nothing but the part of statistical nomenclature. So many of the terms that comprise statistical nomenclatures are familiar. Some commonly used in everyday language like sample, proportion, average, etc. For example, if you talk about that whether the particularly lot is of a good quality or not. So what do? You take a sample from that particular lot and you take the decision whether the lot is a good quality or not. Proportion, what percent of the people like my talk and what percent of the people did not like the talk. What is the average time is taken by the speaker in the conference. So these are nothing but the day-to-day life we are using the nomenclatures, and these are nothing but the part of a summary statistics. Statistics is nothing but it is a science of a decision making. The decision should be taken based on the availability of the data. Many of the time people say that, sir, statistics, we learn statistics in commerce. So statistics is a subject of commerce. No, my dear friends, statistics is a subject of a pure science. We develop a theories and users can use that theory. So they are the applications they are applying in their field, but we are the subject of a pure science. And we always say that it is a subject of a decision making. Why? Because decisions should be taken based on the availability of the data. So now the first question arises to us, what is the data? So data is nothing but it is a collection of information. Now information can be of a two type, either it is a quantitative or qualitative. Next, please. The qualitative data can be kind of a data where you can say that there are no physical measurement tools are there. For example, if I wish to measure the eye color or the types of injury, genders, So there are no physical measurement tools are there. It's a purely attribute. So in short, the difference between the qualitative and quantitative is a data which cannot be classified or quantified. Such data is said to be a qualitative, and data which can be quantified. Such data is said to be a quantitative data. Qualitative data is further classified into the two parts. One is a nominal and second one is a ordinal data. Now, what is the nominal? Nominal data is nothing but it is simply a label to identify or classify the object. For example, the patient numbers, subject numbers. So these are nothing but your social security numbers, your passport numbers, your Aadhaar numbers. So these are nothing but unique numbers to identify the person. When you say that ordinal data, if you associate the magnitude of ordering property with the nominal data, then such data is said to be a ordinal data. Common examples are that types of injury. Now, doctor apply their perception and then identify the injury is minor, moderate, or severe. Types of cancer, preference of selection of doctors. There are 5 hospitals are there, 5 doctors are there. Which hospital should I prefer first if something is happened wrong with me? Next, please. Other type of data, it is a quantitative. As I already said that a data which can be quantified. So such data is said to be a quantitative data. It means some measurement tool is there to measure the data. So such data is said to be a quantitative data. Common examples are height, weight, blood sugar level, cholesterol level. So these are nothing but the part of a quantitative data. Now, quantitative data is also classified in two parts like qualitative data. In qualitative, we have a 2 part is nominal and ordinal. In quantitative, we have a 2 part called interval data as well as the ratio data. So in interval data, difference between consecutive categories is known and equal. For example, if you are measuring the satisfaction level on a 5-point scale, for example, after completion of this talk, your organizer will say that share the views or your responses about the content and quality and delivery about the talk on a 5-point scaling. So let us say 1 means the worst and 5 means the good and 3 is a neutral. So difference between a 1 and 2 is category is same. That is the perception of the participant between the category 1 and category 2 is same. That is the same between 2 to 3. So that means it is saying that if the difference between consecutive category is known and equal, then such data is said to be a interval data. But there is a problem with the interval data is that 0 is not fixed. It is an arbitrary. For example, if I am saying that 0 degrees Celsius temperature, it does not mean that there is no temperature, but one can measure it in the other measurement like in a Kelvin or Fahrenheit. So it means that 0 is not fixed. And that is how there is a need of a development of a new data, which is called the ratio data, in which we say that 0 is fixed and which satisfy all the properties of the previous data, that is nominal, ordinal, and interval data. And common examples are the height and weight. Next, please. Why I have discussed this? The precaution is required before analysis of any data is given to us. Before analysis of any data, one has to first check the nature of data, whether it is a qualitative or quantitative. The reason is statistical techniques were developed with respect to the measurement of scale. One cannot use a statistical technique like t-test, ANOVA for a qualitative response. It is applicable only if response measured on a quantitative scale. Next, please. So, according to me, if any non-statistician who can remember these tables while analyzing their data, I don't think so that they find any comments in the research paper. This particular table is classified into 4 columns. First column is talking about the measurement of scale. Second is nothing but the identify via common examples which statistical techniques can be used. That is the descriptive statistics, and another technique is the inferential statistics. Before going to discuss this table, I would like to talk about it. In statistics, we have one of the branch is called the statistical inference. Now, statistical inference is classified into the 2 parts. One is the estimation and second is a testing of hypothesis. So descriptive statistics is associated with the estimation part, and inferential statistics is nothing but it is talking about the testing of hypothesis. Common examples are, or you can say that if the measurement of level is a nominal. So what are the common example? Gender and patient number. Why? because nominal is nothing but simply a categorical data. So male, female, and transgender. So this is nothing but it is a category. What kind of a descriptive statistics you can apply? Frequency, percentage, and mode. That means how many male have participated in this program, what percentage of the male have participated, who is more, whether the male or female. So that is the mode. So if your data is nominal, you can apply the descriptive statistics like frequency, percentage, and mode. And if you talk about the inferential statistics, we wish to see that whether the test of proportion, that is the P is equal to P naught, whether if I had given some treatment, or if I have plant a seed, so how many seeds are germinated, whether the seed is going to germinate or not. So what is the probability of germinating? So suppose if I am saying that probability of germinating is a 0.7, then how will you assess whether my claim is correct or not? So one has to go for the binomial test. Suppose if you wish to talk about that, there are the 4 different hospitals are there, and you wish to identify what is the average proportion of a smoker across the hospitals. So there are 4 hospitals, and if I wish to compare the proportion level, P1 equal to P2 equal to P3 equal to P4, I have to apply the test, which is called the test of proportion, which is known as a chi-square test. So what I mean to tell you, according to the nature of variable, you have to identify which scale is suitable, and according to the scale, which statistical technique is suitable. If your data is ordinal, whereas what was the association between nominal and ordinal? It is saying that in nominal data, it is simply a categorical. It is able to identify the object. But if you associate magnitude of ordering property, we call them as a ordinal data. So types of injury is a kind of a example, preference of rating, whether the injury is minor, moderate, or severe. So of course category, but associate of ordering properties with that. Common descriptive statistics, you can use a median and percentile. And inferential statistics, you can use the Spearman correlation, or you can say that if you wish to compare the several treatment and where data is measured in an ordinal level, you can use the Kruskal-Wallis test, which is an alternative to the one-way ANOVA discussed by the Fisher, Professor R. A. Fisher. Friedman ANOVA, it is an alternative to the randomized block design, RBD. So if your data is measured in an ordinal. Ordinal means, suppose you have a quantitative data, even you have converted them into the rank. So ranking is nothing but they are talking about the ordinal. Now, if your data is interval, as I said, that common example is satisfaction measured on a 5-point scaling. You can apply the descriptive statistics like mean, variance, standard deviation, etc. And the inferential statistics you can prefer like a t-test, which include the one sample t-test, independent sample t-test, pair t-test. You can use the ANOVA. It is talking about the CRD, RBD, LSD, all factorial experiment, regression analysis, factor analysis, if your data is measured on an interval scale. In ratio, the common example are height and weight. So you wish to deal with the geometric ratio. If you are talking about the growth, so geometric mean and harmonic means are suitable when your data is ratio. But if it is of interval and scale, both, still you can use the inferential statistics like t-test, ANOVA, regression, factor analysis, and descriptive as this mean, variance, standard deviation, and others. Next, please. So this we talk about it is dealing with if you have a univariate data. And above the discussion which table I have given, it is dealing with if you have a univariate data. But now there are the situation that we come across with the one or more variables. Suppose we have 2 variables, like one is a dependent, one is the independent variables. So regression method describes the relation between the response variable and one or more explanatory variables. Usually it is said that regression methods are used with the continuous response. So regression analysis can be performed when your data is a quantitative. Which one? Response. That is called dependent variable. And explanatory variables, it is also a quantitative. So it means that when both X and Y are quantitative in nature, then we can apply the regression model. So common example is that you are talking about the height and weight of individual. So you have to first identify which one is dependent, which one is independent. We know that the height is also quantitative. We can measure. Weight we can also measure. But we know that there is a correlation between the height and weight. Now, correlation is simply saying that whether the correlation is positive or negative, and it is saying that amount of the correlation, but it does not tell that what kind of the change we can make on a one variable and what kind of change corresponding you observe on the another variable. So first thing is that you have to identify whether data is a quantitative. Yes. Now you have to also identify whether there is a logical relationship between the X and Y is possible. So we know that the height and weight, they are logically related. Now among that, you have to identify which one is dependent on which one is independent. So whether the height is dependent on the weight or weight is dependent on height. So we know that as the height increases, there is a chance that weight may increase, but weight increase doesn't mean that there is an increase in the height. It means that height is considered to be independent variables and weight is considered to be dependent variable. So before analyzing this, you have to identify this. Then you have a software is available to you. You can just click it, you can have output and you can have an interpretation. So most of statistical methods we have learned depends on the continuous data. However, we sometimes have a binary response, such as yes or no. Whether if I have given the drugs to the patient is cured or not. If I have given the more hours of a teaching, whether the student has successfully passed or fails. Whether I had given the specific brand of a seeds, whether the seed is germinated or not. So what I mean to tell you that when your response is a binary, failure and success, yes and no, then we cannot directly go with the regression analysis, which we have studied in our BSc level, like simple linear regression and multiple linear regression. When the response are measured with the binary data, it should be treated as a categorical data and the number of response should be counted. So how many seeds are germinated, how many are not germinated, how many patient is cured, how many are not cured. So we have a two type of outcome. Either we have a discrete outcomes, that is the categorical, or we can have a count, that is also a discrete. So when explanatory variables are not continuous, they are the dummy or dichotomous variables, we can not apply the techniques like the regression and ANOVA. On the other hand, when response variable are discrete, taking on a two or more categories, the logistic regression is more suitable. So next table will explain you that which statistical technique should be used under which condition. Next, please. So depending on the characteristics of data, whether it is a continuous or categorical, and the role of the data, that is the explanatory or response, the appropriate statistical analysis method should be chosen. And following table give the quick guideline to choosing the analysis based on the independent and dependent, that is explanatory and response variables. Next, please. So if you look at that, in a column it is said to be dependent, in a row it is said to be independent variables. So let us say your dependent variable is a continuous, that means it is a quantitative, and independent variable is also quantitative. Then which technique is suitable? You can apply the regression. For example, dependent is the weight of a respondent, independent is the height of the respondent. You can apply the regression analysis. In botany, you can say that the height of the species, and it is a function of a time. So time is independent, which is a continuous, dependent is a height, it is continuous. So you can use the growth curve linear models. So these are nothing but the part of a regression analysis. But now suppose you say that dependent variable is a continuous and independent variable is a categorical. Then one has to go for the analysis of variance. So for example, if you are working in a zoology and you are talking about the weight gain by the fish. So there are three different types of the food is prepared by the zoologist, and they divide particularly three groups. One group, they are giving the one food. Second group, they are giving the second food. And third group, they are giving the third food. And what is your response? The response is a amount of a gain in a fish weight. So amount of a gain in a fish weight is nothing but your continuous variable. So response is a continuous, and types of a food, it is a categorical. So my basic idea is that which food is more important, which food is really helpful to increase the weight gain of a fish. So first I will check that whether the all foods are equally effective or not. And to compare the more than two treatment, we can have a technique called CRD, which is nothing but the analysis of variance. And then once the hypothesis is rejected, we can proceed whether is there any significant difference between at least two types of a food? Yes, then which food has a more fish gain weight? So that food will be suggested to the fisheries industry, because fisheries industry is interested in this area. So what I mean to convey, look at the nature of variables and then select the technique. I would like to especially point out here, we are running a certificate course in SPSS, our department. So one of the research scholar from the zoology, he came to me, sir, I have this is the fish weight gain data, and this is my different types of foods which we have prepared for observing the fish weight gain. So which technique should be suitable? I told him, like why I'm discussing the same thing with you, that you apply the analysis of variance and take the decision based on that. He submitted the papers to one of the elsewhere journals, which is in the Scopus, and he got the queries from the reviewer that according to the nature of the data, yes, according to the nature of the data, you have to apply the regression analysis, not the analysis of variance. He came to me, he said, sir, you taught me that we should apply the ANOVA, and now the problem is that the reviewer is saying that you should apply the regression. Now I want to submit my thesis. So should I do the regression and submit as per the suggestion made by the reviewer? I said it is absolutely wrong. You should not go with that. You should ask them, sir, can you suggest me some reference book where such kind of the analysis is done and where the regression analysis is used, so that I will acknowledge that particular refer that book. I will write in the reference and do the analysis, because when I have discussed with the professor of statistics, he said that according to this, we should apply the analysis of variance. However, you are the right. Yes. Any questions? Okay. So, however, you can suggest this. So we observe that in 15 days, he got the answer that your paper is accepted. So what I mean to tell you, that don't blindly look at the paper has been published. If somebody has made the mistakes, you are just copying their mistake in your research paper. So my humble suggestion is that whenever you are dealing with any domain, you should try to understand the nature of variables. According to the nature of variables, try to use the statistical techniques and give your interpretation. Now, suppose your dependent variable is a categorical and independent variable is also a categorical. So you can use the logistic regression analysis. Similarly, if your dependent variable is categorical, it is continuous, you can apply the logistic. If it is categorical, independent is also continuous, categorical, you can apply the log linear models. And even if your response is categorical, but your independent variable is a mix, either categorical or continuous, till you can proceed the logistic regression. Next, please. So now, based on this, you can apply the suitable technique and you can use the software. So I'm not going to discuss much about the R, but R is an open source and it is available easily via Crane website. So you just write to the R in a website, you can easily access, you can easily download. There is no need to work with the commercial software. It's an open source and you can use it. And it can allow you to do the variety of statistical analysis, like you can make the variety of graphs, linear and non-linear modeling, classical statistical text, time series analysis, clustering. You can do the principal component analysis. Many multivariate techniques, you can easily do that. Even the advantage of R is that it can be extended via several packages. So day by day, researcher is creating a new package and they will upload in the R website. So you can easily download and you can use it. Next, please. So this is all I say, that it can be extended via packages. There is no need to even go for the MATLAB, because all mathematics operation you can easily perform using the R language. It has the good advantages that you can import the data from the various DBase. You can take into the R and you can analyze it, and then you can export your output into the presentation mode. Next, please. So these are from R project. You can download the R. Next. This kind of variety of graph you can make: contour plots, prospective plot, pie chart, multiple scatter plot. So variety of the graphs you can easily make. Next, please. So first we talk about the measurement of scale. Then we talk about the choice of a statistical analysis. And the third point was that test of independence of orders. So let us, I have a data of a serum cholesterol changes. That means a patient who came to the doctors, their serum cholesterol level is measured. And after giving the treatment, the patient's serum cholesterol level is measured. And the difference is measured in terms of a percentage. So suppose I have such kind of a data. So many statistical tests assume that observation in samples are independent. So whenever you apply any testing of hypothesis, in statistics, we say that there are two assumptions are mandatory. One is a repetition, that is called replication, and second one is a randomization. So randomization is first. And if you wish to improve the power of your test, then you go for the replication, which can allow you to increase your degrees of freedom. So in other words, in order to see whether the data are collected is relevant or irrelevant, if your data is relevant or irrelevant, in the same set, it is in the form that whether it is a random or not. If the order does not matter, then the sample is not a random, and you cannot draw an accurate conclusion about the population from which the sample was drawn. I would like to here also stress that in statistics, we have two types of designs called sampling designs. One is a probability sampling scheme and another is a non-probability sampling scheme. Most of the people what do? They do the sampling via purposive methods, via quota-based sampling, via convenience-based. So whatever the samples you have taken in your study, you cannot generalize their findings for a population. So whenever you wish to generalize your findings, because as a researcher, whatever we are giving the ideas or whatever the conclusions we are drawing from a sample, we would like to see that whether that sample is a true representative of a population or not. And we wish to infer about the population. But if you are using the non-probability sampling scheme, it means that the samples are not random. You cannot generalize it for a population. Therefore, it is prudent to check the data for a violation of this important assumption. We can use the run test procedure to test whether the order of values of a variable is random or not. The first procedure is that it is classify the value of each variable as falling below or above the some cutoff point. Usually we say that the cutoff point is a median, and then test to ensure that there is no order to resulting in a sequence. Next, please. So this is the serum cholesterol data was analyzed using the R, and you can see that the test statistics value is a 1.45 and the corresponding p-value is a 0.1456. So p-value is nothing but it is a probability value, and we wish to see that whether that particular test statistics is belong to the critical region, that means rejection region, then p-value is always less than 0.05, where 0.05 is nothing but level of significance. That means how much percentage of the errors we are allowing in our decision while taking the decision about this hypothesis. If I'm saying that 0.05, it means I'm talking about the 5%. So if your cutoff point belongs into the critical region, that means the p-value is always less than 0.05 and you reject your null hypothesis. That means you are in the favor of your alternative hypothesis. So in this setup, what is the p-value? It is a 0.1456, which is less than 0.05, suggests that serum cholesterol level is randomly distributed. And what the null hypothesis here is, the data is independent of order. Next, please. Next, please. Yeah. Next, we talk about whether the data is independent of order or not. So first, we have seen that it is independent of order or not. Second is that whether the data is normally distributed or not. Why? Because most of the statistical techniques like t-test, ANOVA, regression, the underlying assumption is that data is coming from a normal population. Normal population means it possesses the normal distribution, or sometimes it is known as a Gaussian distribution. So therefore, it is mandatory that before applying any statistical technique, we should see whether the data is a normally distributed or not. Why there is a need of a assessment of assumption? Suppose if you talk about any building, so what is whether the building is no, don't change it. I will tell whether the building is having a strength or not. So the strength is depends on the base of a building. So similarly, if the base is sound, base is powerful, the building has also a better life. So similarly in a statistics, what are the basic foundation? The foundation is nothing but their assumptions. So we have to see that whether your data satisfy the basic assumption or not. If yes, then you can talk about the assessment of the further test. Otherwise, you have to look for the some alternative approach. So null hypothesis here is that data is coming from a normal distribution. Look at the p-value. What is the p-value? It is a 0.84, which is greater than 0.05, suggests that data fits the test distribution. And here we assume that the distribution of the test is normal. Hence, we say that serum cholesterol changes in percentage for patients after administration of a drugs follow the normal distribution. Next, please. So when you talk about the testing of hypothesis, there are two types of a testing of hypothesis. One is a parametric test and second one is a non-parametric test. Now, when you talk about the parametric test, there are various tests are there. One sample T test, independent sample T test, pair T test, analysis of variance, which include the CRD, RBD, LSD, that is the two-way ANOVA. And you can also talk about the factorial experiment. While in non-parametric, we can have a sign test, Wilcoxon sign test, Mann-Whitney, U test, Kruskal-Wallis test. So these are the different tests are there. But which tests are parametric and under which condition we should use the parametric, under which condition we should use the non-parametric, which test is alternate to non-parametric test is alternate to the parametric test. So that idea I will try to narrate you. Next, please. So in testing of hypothesis, these are the, if you look at the any book, such kind of a nomenclatures you can observe. Mu is nothing but population mean, sigma square is the population variance, tau I is nothing but ith treatment effect, mu I is nothing but population average of ith population, Yij is nothing but the response of the jet subject on a ith treatment, p value is nothing but probability value, h0 is null hypothesis and h1 is alternative hypothesis. Next, please. So when you apply the testing of hypothesis, you require to know the certain things. So a statistical test of hypothesis is a rule or procedure which makes one to decide about the acceptance or rejection of a hypothesis. To test any hypothesis about the population parameters, we should require 4 points. One is we need a distribution of a test statistics. For example, if you are testing the mu equal to mu0, that is the average value of a population value is a mu0. That is mu0 is nothing but assigned value or the non guess value. If you wish to apply this one sample problem, there is a test called the one sample T test. So you require the test statistic, that is the one sample T test. What is the distribution of that T test? It is a T distribution. Then you require the level of significance because you wish to take the decision whether the hypothesis should be rejected or accepted. So how will you decide? So it is saying that you allow the errors in your decision. So how much percentage of errors are you allowing? So if you are saying that 5%, so your level of significance is a 0.05. And you are talking about the decision, whether the p-value less than 0.05, reject at not, p-value greater than 0.05, accept at not. So there is a difference between the research or statistical hypothesis or scientific hypothesis. The research or scientific hypothesis is the hypothesis which a researcher postulates to meet his objective, whereas statistical hypothesis is testable formulations of research or scientific hypothesis or the functional relation about the parameter. So you are simply giving the statement in a research hypothesis, but when you convert that research statement into the some formula, so then it is said to be a statistical hypothesis. Next, please. So now the question is that what should be the null hypothesis? So according to Fisher's, null hypothesis tested for its possible rejection. So any hypothesis which has the possible chance to reject is called the null hypothesis. Why? Because we are never interested in the null. We are always interested in the alternative. An alternative hypothesis is a statement about the population parameters which provides an alternative to the null hypothesis and the range related values of the parameters. So again, it is in the range. So it is an alternative to the null hypothesis. Next, and it is denoted by H1. Next, please. So parametric test. When you talk about testing of hypothesis, you should know what is the null hypothesis. So as I said that a hypothesis which has the possible or a statement which has a possible chance of rejection, you should frame them as a null hypothesis. Once you identify the null hypothesis, you should know that it is a single one population problem, two population problem, or a K population problem. According to you, decide the test statistics. Once you have a test statistics, try to identify the distribution of a test statistics, apply the level of significance, and take the decision. So in this setup, there are two types of a hypothesis called the parametric test and non-parametric test. So parametric test requires the assumption about the population from which the sample has been drawn. That is the form of a probability distribution is assumed. So when you wish to apply the parametric test, it is mandatory that you should know that how the data are generated and what is the patterns of generation of a data. That means what is the distribution of a population. As I said in earlier discussion, when you talk about the probability sampling design and non-probability sampling design. So probability sampling design means there are certain methods via that you can select your random sample from a population and you can generalize your findings. Also, this test is applicable to data which are measured only on an interval or ratio scale. If your data is measured on interval or ratio scale, it means if it is of a quantitative in nature, you can apply the parametric test. If this assumption do not hold good, we do not meet the requirement of parametric test. And still, if we use this test, we will not get reliable testing procedure about the population parameter. So many of the non-statisticians, because of the softwares are available, they have the data of whether the patient is cured or not. It is measured on a 1 and 0 scale, and they say that, sir, we applied the we have find out the mean of the average curing of the patient, and we apply the one sample T test. Now, I don't know that why they are applying that, because it is whether the patient is cured or not. So when the patient is cured or not, it means the data is a categorical. That means what percentage of the patient is cured and what percentage is not cured. That means you are talking about the test of proportion. So test of proportion, you have to apply the binomial test. One cannot use the one sample T test. So what I mean to tell you that you should understand the nature of variables, see whether the findings has the feasible interpretation or not, then you can select the test. And still, if you can use, you cannot give the reliable estimate about the population parameters when there is a violation of the assumptions. Most popular parametric tests are nothing but one sample T test, independent, two sample T test, pair T test, and analysis of variance. Next, please. Next, please. Yeah, non-parametric test. So alternative to the parametric, you can have a non-parametric. In real practice, it is very difficult to meet the requirement of a distribution of a test statistics. Due to this stringent assumption of a parametric test, distribution of a test statistic required, we have to search an alternative testing procedure which do not require this kind of a stringent assumption. That means suppose your data is there, and if you don't want to have a distribution of a data, then we cannot apply the parametric test. So what alternative test we can apply? It is known as a non-parametric test, where non-parametric test is a test which is a free from distribution. It is also known as a distribution free test. Further, this test is applicable to all types of data, whether it is a nominal, ordinal, interval, or ratio. As I say that if your data is a quantitative, we convert them into the rank and then we can use it. So non-parametric tests are mainly tests based on the ranking. The following most popular tests are it is not part of parametric test, it is a non-parametric test. So test is a binomial test, chi-square test, binomial, as I said, that the test of proportion. When you wish to compare the several proportion, you can use the chi-square test. Even when you wish to see that is there any association between two attributes or not. For example, is there any association between the cardiovascular disease and the diabetes? If I wish to see that whether the diabetes is a risk factor for a cardiovascular disease or not, so I have to see that is there any association between these two attributes, and one has to go for the chi-square test for a contingency table. Mann-Whitney test, it is an alternative to the independent two-sample t-test. Wilcoxon sign test for a related sample, it is an alternative to the parity test. And Kruskal-Wallis et test for a k-independent sample, it is an alternative to the analysis of variance, that is called the CRD. Next, please. So we will see this one sample test problem. It is the data is of average potency. The average potency a pharmaceutical person can easily understand that what is the potency of a drug. And the potency of the drug is collected from a population, from a sample, and we find out the average potency. So we will consider an experiment to assess the effect of a change in manufacturing process on the average potency of a tablet product. So whether if I do the change in the manufacturing process, my aim is that whether the average potency of the tablet is changed or not. Now we will tell whether the average potency is higher is better or below is better, a person who is working in the pharmaceutical industry. So whether the higher potency is better or lower potency is better. So they will tell you. Our objective is that we have some data, and it has been asked you that our average potency will be this, and we wish to see whether the manufacturing processes provide this average potency or not. So what the study is here, from the literature review, it is found that the average potency of the drug is a 5.01 mg. So it is a single population problem because you are talking about the average potency. So it is measured in a continuous scale, and it is saying that the 5.01 mg is a true process parameters. So their aspect is they wish to test whether the H naught mu equal to 5.01 mg or not. So it is a single population problem. Mu is a population parameters. 5.01 is a assumed value of a population parameters. And to test this, one has to go for the literature. In literature, it is saying that you can use the one sample t-test problem. The new batch was made with a modification of a usual manufacturing process. 20 tablets were assayed and results were collected. That means randomly they have taken the 20 tablets, they do their bioassay and they find out their potency level, and then they see that whether the modification is giving the average potency is a 5.01 mg or not. So our objective is to determine if the process modification results in the change of average potency from process average of 5.01 mg or not. That means the value of mu under the null hypothesis is 5.01. So before performing the t-test, first we have to see whether the data is independently distributed and data is normally distributed. Why it is required? Because as I said that the foundation is very important. If you violate the foundation, then the strength of the building is not the good. So whenever you wish to see that I wish to apply the parametric test, it's a one sample t-test, I have to verify the assumption. Next, please. So look at the particularly the p-value corresponding to run test. It is a 0.818, which is greater than 0.05. We suggest that the data is independently distributed. But when you look at the graph, this is the histogram and second graph is a normal probability plot. So histogram is look like as a positive skewed. Normal probability plot is saying that all the point is not covered by the line. It means the data is not normally distributed. So graph suggests that data is not normally. Run test say that data is independently distributed. So we will apply the test. So there are two tests is that one is Kolmogorov-Monro and second test is in R available is called Shapiro-Wilks normality test. So that will tell you whether the data is really normally distributed or not. Graph does not support your assumption. So we will see the test of hypothesis. Go to the next. So look at the one sample Kolmogorov-Monro test. If you standardize data, it is saying that the standardized data follows the normal distribution because the p-value is 0.8851, which is greater than 0.05. Or you look at the Shapiro-Wilks normality test, which is the inbuilt test is available in the R. Their p-value is a 0.08, which is also suggests that it is a greater than 0.05. We can say that data follows the normal distribution. So now first two assumption is verified statistically. Run test is saying that data is independent of order. And Shapiro-Wilks normality test is saying that data is independent of, data is normally distributed. So first two assumptions which are required, which is satisfied. And now we can proceed to test the hypothesis at naught mu equal to 5.01 mg. That means the average potency of the drug is a 5.01 mg. Now, which test is applicable? One sample T test. So go to the next slide. Look at the output of a one sample T test. What is the value of a T statistics? It is a 3.08. Total, there are 20 observations. So n minus 1, that is a 19 degrees of freedom, and p-value is a 0.006. Now this p-value is less than 0.05 or greater than 0.05. So it is a less than 0.05 because I am not saying 0.05. I am saying that 0.505. So it is a less than 0.05. So we say that the null hypothesis is rejected. And what is the alternative? Alternative is saying that it is not a 5.01. Further, the R is also giving the confidence interval 95%. With confidence interval, we say that the average potency of the drug lies between 5.02 to 5.10 mg. And what is the estimated value of the potency level? So we call them as X bar. X bar is nothing but the 5.0x. What is X? X is nothing but the potency of a drug. How many potency we have observed? 20. So x1 is some value, x2 is some value. How many we have? X1, x2, x20. Their average value is nothing but X bar, and that X bar value is a 5.06. So look at that 5.06 is greater than 5.01. And that is how the value of a T statistics is positive. So the value of a T test, which is positive and hypothesis is rejected, it is indicating that the average potency of the drug is not 5.01, it is higher than the 5.01. So from this analysis, we can have a following conclusion. From the above table, we can say that the average potency of the drug is 5.065, having standard deviation is 0.085. Okay, 0.085 I have not shown here. The p-value corresponding to T test is 0.006, which is less than 0.05, indicates that our hypothesis is rejected. Which hypothesis? Null hypothesis. It means that average potency is significantly different from the average assumed value 5.1. So we statistically say that it is significantly different from 5.01. Further, the value of a T test is 3.081, which is greater than 0, suggests that the average potency of the given data is more than the assumed average potency. And that is what we can see, that 5.06 is greater than 5.01. It helps us to take the decision that modifying manufacturing process of drug is good compared to the older one. So if you talk about the person who is working in the pharmaceutical domain, they will simply say that, sir, the average potency of the drug is improved. It is better for the manufacturing process. So it means that whatever the modification we have made in a manufacturing process is considered to be a good, and we should go with the modified processes. So next, please. It's a thank you. But before saying a thank you, I would like to also convey that when you apply the parametric test, so whenever there is a possibility, apply the parametric test, because parametric test is considered to be most powerful compared to the parametric test is more powerful compared to the non-parametric test. So whenever there is a possibility, apply this. Now, I think a few questions are there I had observed. How to choose the best model? What are the tests used for statistical analysis, and what are assumptions required for a regression analysis? Okay, if you are dealing about the regression analysis, as I say that the both y and x should be continuous in nature. That means they are quantitative in nature. So before applying the regression, first you have to see that whether the both variables are quantitative in nature. Second, is there any logical relationship exists between two variables or not? Then you have to see whether there is a significant correlation between them or not. Once you identify this, then you have to go for the assessment of a basic assumption of a correlation. First assumption is that the response, we have the assumption is that y is equal to beta 0 plus beta 1 x1 plus epsilon. So we had an assumption on an error. So error should be randomly distributed. Error should be normally distributed. So you fit the regression models, obtain the residuals, that is the estimate of errors. You see that whether the residuals are independently distributed, apply the run test. You see that residuals are normally distributed, apply either Shapiro-Wilks normality, or you can apply the Kolmogorov-Smirnov test. Once it has been identified, you can proceed to fit the final models, in which you have to perform the two types of a test. One is a F test, that is the simultaneous testing of parameters beta 0 and beta 1 are 0. Once the hypothesis is rejected, it means that whatever independent variable you have selected, for example, height and weight. So weight is a function of a height. So height is important to predict the value of a weight. Then you look for the individual testing of a regression coefficient. You apply the t-test, and whether your model is a good or not. So you have to apply the goodness of fit criterions. One of the criterion is a R square. R square is nothing but it is a square of coefficient of determination, or it is a also known as a percentage of the variation of a y is explained by the independent variable. So if he is saying that if R square is very high, the maximum value of R square is 1. If you say that R square is more than 0.9, whatever the variables we have selected in our model, that variable is really helpful to study the response variable. But there is a demerit of a R square also. If you add one more variable, R square is saying that it is always increasing. If you add variable, what saying that R square always increasing by adding the variables. But now the question is that whether the added variable is important or not. That is another added variable. To see that, one has to look for the adjusted R square. And the adjusted R square is saying that if the added variable is not significant, adjusted R square is decrease. R square always increase. Whether that variable is important or not, if you add, it is saying that we are explaining why. And that's why to see that whether the second added variable is important or not, one has to see the adjusted R square. If R square and adjusted R square both are increasing by adding the another variable, let us say x2, then we say that x2 is important to know the value of y. But if R square increase, adjusted R square decrease, it is saying that added variable x2 is not important and we should not allow in our model. There are further criteria also is that one should look for the AIC and BIC. AIC is nothing but the archaic information criteria. BIC is called the Bayesian information criteria. If lower the value of AIC, lower the value of BIC, whatever the models we have taken for a comparison among the several models. So lower AIC, lower BIC, better the models, that model should be selected. Then one should also look for the mellow CP criteria. Lower CP and CP equal to P. P is nothing but number of independent variables in the model. Then we also say that the final set of regression where the lower the CP is better the model. So I think I should end up to here. Any questions, anyone? And I'm very happy to answer if you have. Because this I had observed, most of the non-statistician has the problem related to selection of a technique. Softwares are there, they can analyze it.
In his lecture, Dr. Reykundaliya has touched upon varied aspects of statistics, right from the basics like measurement of scales to the more advanced concepts like inferential statistics, testing of hypothesis, p-value, and ANOVA, with absolute ease and smoothness, such that even someone not well versed in statistics could follow it with a good level of understanding. Your lecture was extremely informative, and with appropriate examples, you made it interesting as well. With these words, I thank Dr. Dharmesh Reykundaliya sir profusely for his excellent lecture. We are extremely honored and obliged for the same. Thank you, sir.
Thank you, ma'am. Thank you.
The reins of the conference to Dr. Hina Pathan for conducting the second session of the day.
Thank you. Thank you, ma'am.
A very good afternoon to our respected principal ma'am, Dr. Swati Wawal, co-convener of the conference, Dr. Bhavita ma'am, members of the organizing committees, my fellow professor colleagues, and all young minds. I, Dr. Hina Pathan, Assistant Professor, Biotechnology Department, feel privileged to warmly welcome resource person for our session, Dr. Anukul Shenoy. He is currently an instructor of medicine at the Pulmonary Center in Boston University School of Medicine. He has research interests focused on understanding how lung epithelial cells act as a critical coordinator of lung immunity during health and disease. Although not destinated at USA, Dr. Anukul started his journey from Mumbai University, completing BSc and MSc. In no time, his strong ambitions and dedicated hard work made his dream come true to pursue Ph.D. from USA in microbiology and immunology. He started his PhD work at University of Texas Health, and before moving with Dr. Ori Wheeler to the University of Alabama, where he worked on host pathogen interactions during Streptococcus pneumoniae infections. After receiving his PhD degree in 2017, he completed his postdoctorate in the pneumonia biology with Dr. Joseph Misgard at the Pulmonary Center in Boston University. His current research interests are focused on understanding the crosstalk between the distinct lung epithelial cells and CD4 T cells in context of infection, inflammation, allergy, and immunity. Dr. Anukul has published various new research papers and articles in international journals and has been a part of prime member in American Association of Immunologists, American Thoracic Society, American Society of Microbiology, to many more. Moreover, with this excellence in research, Dr. Shenoy has few honors and awards under him, namely ATS Pulmonary Infection Science Award, the American Association of Immunologists Young Investigator Award, and many more. In the past too, he has been a reviewer and has presented his work at numerous international conferences. An interesting fact about him, apart from his research interest in the field of mucosal immunology, Dr. Anukul is a pioneer national swimmer, winning more than 250 gold medals at various international and national levels. His contributions and achievement at such a young age is lengthy, but owing to the limitation of time, I now hand over this session to Sir, as everybody is waiting enthusiastically to hear from him. As today's conference on A Tale of Two Cells, Lung Epithelial CD4 T Cells, Dialogue in Health and Disease.
Awesome. Can everyone hear me? I just wanted to make sure that people can hear me and people can see the slides. If someone can give me a thumbs up, that'd be great.
Sir, we can hear you.
Okay, excellent, excellent. So, okay, awesome. So thanks for this really generous introduction. So as one quick fact correction is I used to be a swimmer. I don't swim anymore. My current research life doesn't accommodate me practicing as much as I used to back when I used to do my bachelor's and my master's back in the University of Mumbai. But today I've thought I'd share a couple of research stories I've been working on as a postdoctoral fellow and now as an instructor in the Boston University School of Medicine. And these are about two of my major cell types. These are the cell types I love and cell types I think about in my dreams and my sleep. So it's the lung epithelial cells and CD4 T cells. So it's going to be a slightly immunology heavy focused talk from here on. So let's see if I can get this moving. Okay, great. So we all understand that epithelial cells act as a direct interface between the core of the body and the external environment. So if you look at any epithelial tissue like the conducting airways or the alveoli or the intestine or the skin or the nasal epithelium, the epithelial cells are literally the cell type that is exposed to the external environment and is protecting the inside of our body from the exposed environment. And given its location, these epithelial cells are constantly bombarded with different types of microorganisms. Some of these microorganisms are actually commensals like E. coli in your intestines, versus some of them are pathogens that can actually cause disease. In addition to that, these epithelial cells, considering our current modern environment, is constantly dealing with a bunch of physical and environmental stressors, a bunch of wounds, allergens, and these epithelial cells are hence required to constantly sense these external signals so that they can talk to the immune cells underlying this tissue so that they can launch a good immune response in context of an infection. But what is really interesting that people don't really think about is that in addition to inducing inflammation and an immune response, these epithelial cells are also the stem cells of that particular organ. So once the injury is resolved, the same cells actually have to undergo proliferation, division, and then differentiation to cause tissue repair, which again needs to be regulated, because if that gets dysregulated, then we have diseases like cancer. So given the multifunctional, multi-pronged biology that epithelial cells hold in context of tissue immunity, a large part of my postdoctoral work and my work from here on is being focusing on epithelial immunology. So among a bunch of the immune cells that underlie a tissue, one immune cell that I really want you to focus on for my talk today and take home with you is the specific and newly discovered subset of CD4 T cells called as tissue resident CD4 positive resident memory T cells. So the immunologists usually like to call them as TRM cells, and I'll be calling them the same from here on. What is interesting is if you look at all the barrier organs like the lung, the reproductive tract, the skin, the intestines, and the lymph node, there are a bunch of immune cells that circulate between all these organs, but there are also immune cells that actually stay in this organ and never leave that tissue. And these cells are called as the tissue resident memory T cells. So for the non-immunologists in the audience, I'll give you a brief introduction as to why these resident memory T cells are so new and so different. So our classical textbook tells us that T cells usually move or circulate from the blood into the non-lymphoid tissue or the NLT. From there, it goes into the lymph, then into the secondary lymphoid organs like the lymph node, and then from the lymph get back into the blood. So this is what we think of a T cell as causing immune surveillance, is it keeps circulating between a bunch of organs so that they can maintain its immune surveillance and protection. However, about a decade back, a group discovered that there are some subsets of memory T cells that once it gets into the tissue, it actually never leaves the non-lymphoid tissue. It literally stays there for the rest of its life. And hence these T cells are called as tissue resident memory T cells. And as far as what is known about these T cells, once they park themselves in an organ, they never actually leave, and they do not have an external source that constantly replenishes them. So what we think as a field is that these T cells are actually proliferating in situ in their organ, and they don't probably leave the tissue. So they're essentially fated to death in that same tissue. But what is really interesting is these T cells actually share some innate-like properties. So they act as adaptive immune sentinels in the epithelial tissue. So for example, if it sees its pathogen in the tissue, these T cells can now undergo proliferation, produce a bunch of cytokines and chemokines to bring in other immune cells so that they can fight this pathogen. If the epithelial cell is infected by a virus, for example, SARS-CoV-2, then these T cells can actually kill these epithelial cells and prevent SARS-CoV-2 from proliferating further. As a result of which, these T cells have become very important and a large focus of vaccine immunology and barrier tissue immunology, considering that these epithelial cells are constantly exposed to the environment. And given that these cells are constantly talking to the epithelial cells because of their close surface interaction, I'm interested in understanding how these cells talk to each other in context of an infection. And given that I come from the pulmonary center, the organ of my choice to study epithelial T-cell interaction is the lung. So I don't need to tell people here that lung is one of the most important organs in our body. It is required pretty much to keep us alive because it is responsible for the oxygen exchange and keeping our blood oxygenated. One thing that is really interesting about the lung is it has an inverted tree-like branching structure. So the lung moves from the trachea to the bronchi and bronchioles and the alveolus. And different parts of the lungs actually have different types of epithelial cells that coat the lungs. So when we think of epithelial cell, we usually think of just one type of epithelial cell that essentially acts as a barrier tissue for all the organs. But for the lung, that's not really the case. Different parts of the lungs are actually covered by different types of epithelial cells to do different jobs. So for example, if we look at the bronchi, so if I take a transmission electron microscopy image of a bronchi, you'll see there are two major subsets of epithelial cells. There are these epithelial cells marked here in C that has these tiny secretory vesicles in them. These cells are called as secretory epithelial cells or club cells. And their job is to constantly produce antioxidant enzymes, constantly produce mucus that essentially helps to trap anything that we are breathing into in the airways. And then beside them, there are these cuboidal cells that have cilia on their surface, which are called as the ciliated cells or the multiciliated cells. And the job of these cells is to constantly beat in the same direction so that anything that is trapped in the mucus actually is moved up through the trachea and actually discarded out of the lung. You can imagine, considering the amount of bacteria and viruses and smoke and particulates that we breathe in, these two cells become very important in making sure that our lower respiratory tract actually stays sterile. And then this cell types down in the alveoli here is where the gas exchange takes place. There is an alveolar type 2 cell, which is a cuboidal cell that you're seeing here, whose essential job is to constantly produce surfactant lipids all the time. And why do we need surfactant lipids? So you can imagine the lung is an organ that constantly expands and relaxes as we breathe. If there is no lipid coating on these lung epithelial cells, you're literally going to rip those cells apart every time you breathe, and that's going to kill us. So these alveolar type 2 cells that you're seeing here, their job is to constantly produce the surfactants so that they can maintain the pliability of the lung every time we are breathing. And then there are these really, really thin alveolar type 1 cells, which essentially have a thickness of less than 1 micron. And so they are so thin that oxygen literally diffuses across this epithelial cell surface. And if you can think about this, it's literally this 1 micron thin layer of epithelial cells that is protecting you from all the air you're breathing in from the external environment. So you can imagine how immunologically critical these epithelial cells are in maintaining the airway environment. So a fun fact is if you take the lung epithelium and lay it one beside each other on a flat surface, it literally covers the area of a tennis court. So every breath you're taking, you're breathing in air enough to cover a tennis court worth of surface area. And now you can imagine how important your epithelial cells are in protecting you from your external environment. And obviously, considering the type of air we are breathing in a lot of pathogens. And these pathogens have a tendency to cause a disease called pneumonia. So we all know of pneumonia as accumulation of inflammatory fluid in the airways, which makes it difficult to breathe. And it is actually the leading cause of infectious deaths in the elderly. So if you look at the age of individuals on the x-axis, the case of hospitalization of pneumonia in red, and the cases of death because of pneumonia in blue, what you see is two interesting facts. Number one, incidence of pneumonia is really high in the young children and in the elderly. But what is more interesting is the incidence of death actually increases only in the elderly. So it is a major cause of death in old individuals. And what is interesting is during this young adulthood, between the age of 10 to about 50, us young individuals seem to have this protection phase or a protection window where we appear to possess immunity to these agents that cause pneumonia. And our lab is interested in understanding what is going wrong in these aging adults that they cannot respond or they are failing to respond to the infections. And I, because I'm more of an immunologist that likes to bring a real life perspective, like to switch the question and ask, what is going right in the young adults? So if I give you a broken watch and ask you what is going wrong in the watch, you probably want a watch that works well to compare it to. And this is what I mean by wanting to understand what is going right in the young adults. And my hypothesis is that as children, these early life exposures we are having as soon as we are born, where these babies are taking toys, putting it in their mouth, covering this toy with the bacteria, passing on to the next child, and the next child is putting it into their mouth. They're passing on these bacterial infections from each other. And anyone who has kids here knows that these early life infections for kids are very important in the way they develop their immune system. So our hypothesis is that these early life exposure to microorganisms that are being had in this childhood are very critical to provide this naturally acquired immunity in young adults. So all this is anecdotal and in humans. How do I prove this in mice? So to do this, what we do is we take a pathogen called Streptococcus pneumoniae. So Streptococcus pneumoniae is a diplococcus. It has this really thick capsular structure that you can see on an electron microscopy around it. And this bacteria is the leading cause of bacterial pneumonia worldwide. World Health Organization has put this bacteria as one of the most concerning bacteria to think about. So what we do is we take mice in our lab and challenge these mice with non-lethal infections of this bacteria, essentially to mimic these early life infections these children are having that doesn't really kill the baby. And then we essentially rest the mice for about 4 weeks to really mimic the baby actually growing into young adulthood. And once that happens, then we challenge the mouse with a lethal strain of the bacteria. And this strain is actually so lethal that it will actually kill the mouse. So what we do see is that when the mice have experience with saline versus different strains, we call them heterotypic immune mice, you see that within 7 hours of infection, mice that have had previous experience with different bugs actually see a very strong neutrophilic response in the lung within 7 hours. And this quick neutrophilic influx actually translates to a faster bacterial clearance compared to the mice that had saline experience, and hence they cannot clear the bacteria. So here what we are showing is that people or mice that have had previous experience can actually clear the bacteria much better compared to the mice or people that have not had previous experience. And what we show here is that challenge with this lethal strain actually kills the mice that have no bacterial experience, but the mice that have had an experience can survive these infections very easily. So to understand what is driving this protective response, what we found was that if we give experience only in the left lung of the mouse, so in the mouse, if you infect only the left lung surgically and not the right lung, we see that the left lung of these experienced mice have a specific subset of CD4 T cells marked here in red, that the right lung of that exact same mouse does not have those T cells. And this should take you back to that slide where I showed you that there are specific T cells that sit in that organ and never go into the circulation, as a result of which the right lung of that exact same mouse does not have those T cells. And obviously the mice that have never had an infection, so the mice that were treated with saline, do not have these T cells either. Consistent with these T cells responsible for driving the protection, if you infect these mice that have only left lung experience with bacteria from the trachea, so both the lungs get infected, the left lung actually clears the bacteria, whereas the right lung does not clear the bacteria, suggesting that these T cells are very critical in driving this regionalized protection. And consistent with them driving a robust neutrophilic response, if we take the T cells out and activate them in a plate, these T cells coming from the experienced mice produce a lot of this cytokine called IL-17, which has been known to correlate with a strong neutrophilic response. So is this T cell important? So if I give an antibody that depletes the T cells from these mice and then challenge the mouse with the lethal challenge, you see that the mice that have gotten this depletion antibody cannot protect themselves anymore, suggesting that indeed these T cells are indeed important in protecting these lungs, suggesting that previous exposure is inducing an IL-17 producing resident memory T cell in these lungs. But then I joined the lab and I was really interested in understanding how are these T cells protecting the lung. We know they are producing IL-17A, we know they are there in the lung, But how is this IL-17 protection translating to a faster neutrophilic response? So to ask this question, I decided to do a very simple experiment. I took the mice, challenged them with the infections like I mentioned to give them bacterial experience like young children would have, and then depleted the T cells after 4 weeks. So essentially the mice have gotten infected, they have a lung resident immune response, and then I take out all the T cells out of those lungs and now challenge the mice with a lethal strain and see whether the T cells are required for the neutrophils to come into the lung. And if yes, then what other factors that we know drive neutrophilic response is affected. And what we saw was that consistent with our hypothesis, depleting the T cells using GK1.5, which is an antibody that specifically depletes CD4 T cells, we have a reduced number of neutrophilic influx in the lung. And among all the chemokines that are responsible for calling the neutrophils in the lungs, the chemokine CXCL5 was reduced when we took out the CD4 T cells. So much so that if we actually do a correlation for the CXCL5 levels in the lung and the neutrophilic response in the lung, we see that they are perfectly correlated. So if a mouse has a higher amount of CXCL5, it has a higher amount of neutrophil. But if you take out the CD4 T cells by GK1.5, the mouse has a lower amount of CXCL5 and a lower amount of neutrophil, suggesting that it probably is indeed the case that CXCL5 might be driving the neutrophilic response in this lung. And this was really interesting to us because long time back, our lab had shown that among all the different cell types in the lung, the epithelial cells in the lungs are actually the major sources of CXCL5. So what this data is suggesting to us is the T cells are not directly calling the neutrophils via IL-17, but T cells somehow are probably telling the epithelial cells to produce IL-17, to produce CXCL5 and call the neutrophils in the lungs. So to check if this was indeed the case, we now perform the same experiment, but this time after the lethal infection, we sorted out the epithelial cells. So we performed flow cytometric sorting of the epithelial cells and isolated RNA from them, and then did qRT-PCR to measure the levels of CXCL5 in the epithelial cells. And consistent with our hypothesis, epithelial cells from this experienced mice produced a large amount of CXCL5. And although this data is pretty interesting in itself, the implications of this are really major. What this is telling us is the epithelial cells from a baby who has never had any bacterial experience will respond very differently to an infection compared to the epithelial cells from you and me as young adults, because we have had an immune conditioning to our lung epithelial cells. And this was really interesting to us because this is suggesting to us that the epithelial responses are actually being modified as we are growing older. And this response was not like a complete hyperinflammation of the epithelial cell, because other immune molecules like GM-CSF and CCL20 were unaffected by this experience, suggesting that the T cells are very specifically pulling strings on epithelial cells to produce only CXCL5 out of them. And this suggested to us that there might be a lot more remodeling or reprogramming of epithelial cells that is happening, which we are missing by using qRT-PCR, where we are actually targeting and looking for a particular transcript. So to determine how much of the epithelial cell is actually different, we did the same experiment. And this time we actually did mRNA profiling using RNA sequencing. So we could look at all the RNA being expressed by the epithelial cells. And we had 3 different groups. We had the first group, which got saline experience, which means that this is the equivalent of a baby that does not have any sort of experience. The second group is experienced mice, but they got an isotype antibody. So these mice have got infection and they have their CD4 T cells in the lung. So this is the equivalent of an adult lung epithelial cell like you and I would have. And then the third group is experienced, but before the final challenge, we took out the CD4 T cell, suggesting that this would be the equivalent of a patient who got chemotherapy and their T cells have been depleted from their body. And then we challenged these mice with the bacteria and then looked for the RNA being produced by the epithelial cells. And from the first look, this is already really interesting because this is telling us that at the whole transcriptome level, the epithelial cells of a young baby or an inexperienced mouse responds completely differently to an experienced lung epithelial cell. And a lot of this experience, for example, a lot of this response, like in the module 2 here, goes from red to blue, and then you take out the CD4 T cells and it goes back to red, suggesting that you need CD4 T cells in the adult lung to be able to tell the epithelial cells what to do. And CXCL5 was one of these molecules involved here because it went from down to up and back down. When we looked at the different transcription factors that could possibly be involved, we found NF-kappa B relay, which is most obvious factor involved in immune responses. But deletion of NF-kappa B relay actually did not have any effect in our response, which made us think out of the box for another possible mechanism driving this response. And this is where we stumbled on this finding. We know that TNF-alpha, when it induces inflammation, actually leads to the production of a large amount of pro-inflammatory cytokines and chemokines. And because an immune response always has to be transient, there are actually destabilizing proteins that bind to AU-rich repeats in the mRNA of these chemokines. And what this does is it targets this mRNA for degradation. However, in the same cocktail, if you add IL-17A, the balance gets shifted towards a stabilizing response, where now there are stabilizing AU binding proteins that binds to this chemokines and actually stabilizes that chemokine to produce a lot more of that protein. So we wondered if the T cell that is producing IL-17A is having the same effect on the epithelial CXCL5. And that was indeed the case. If we grow epithelial cells on a plate in vitro and expose them to these different chemokine cocktails, and then measure the amount of CXCL5 response over time, we see that addition of IL-17 actually keeps the CXCL5 mRNA stable for longer, whereas in absence of IL-17, this mRNA actually gets degraded. And this biology is not applicable to the other chemokines, suggesting that the T cells are post-transcriptionally stabilizing CXCL5 and affecting epithelial biology, which was really interesting because we usually think of immune regulation at the transcriptional level and not at a post-transcriptional level. And I have been telling you that CXCL5 might be driving the neutrophilic response, but I have not conclusively proven that to you. And I wanted to prove that conclusively. So what we did was we gave mice bacterial experience and then gave an anti-CXCL5 antibody so that now the CXCL5 is completely depleted in this lung. And what we could see that as soon as the CXCL5 response went down, the macrophage numbers were completely unaffected, but the neutrophil response went down. One very important thing to note here is the T cells in these lungs are completely intact. So what this means is that you could have all the CD4 T cells in your lung, but if your epithelial cells cannot produce the right amount of CXCL5, the lung is as good as unprotected. So when people argue that T cells are the major immune players in an organ, this essentially revisits that hypothesis and argues that T cell might be instructing other cells to do their job, but these other cells might be more important. And if that is the case, I should be able to deplete the CD4 T cells and add CXCL5 into the lungs and essentially protect the mouse. And that is exactly what we saw. When we give these mice experience, the neutrophil response goes up. We take out the CD4 T cells giving GK1.5 and the neutrophil response goes down, as I have been saying so far. But once we add recombinant CXCL5, the neutrophil response comes back up, suggesting that it is the epithelial-derived factor that is responsible for protecting the lung in context of an infection. So hopefully with the first part of my talk, I've been able to convince you that the CD4 T cells in our lung via IL-17 can actually stabilize specific mRNA transcripts. And this allows the epithelial cells to produce more of this chemokine to bring in the neutrophils in the lung and fight the pathogen. So while I've been telling you all this story so far, one thing most people sitting in the audience might be thinking is, oh, then T cells are the king of the hill because they are telling the epithelial cells what to do in a healthy lung. So that was something that I was really uncomfortable with because I always like knowing two sides of the same story. So I essentially asked the next question is, can the epithelial cells in turn talk to T cells and tell them what to do? And the reason for asking this question is that if you remember this particular figure in my third slide in the beginning, In the alveoli, there are these alveolar type 2 cells, which I mentioned are the cells that constantly produces surfactants and keeps the lung from tearing itself open. These alveolar type 2 cells across multiple terrestrial mammals, so across rats or humans or mice, for multiple decades now, have been known to highly express MHC class 2. And these cells are down here in the alveolus. So for people who don't know why MHC class 2 becomes so interesting, I'll give you a quick revision on antigen presentation biology in the field of immunology. So antigen presentation is a mechanism by which the innate immune system actually gets bridged with the adaptive immune system. And this pathway is very important for us to develop immune memory. So every time we get vaccinated, the innate immune cells, a particular subset of them, have to present that vaccine antigens to an adaptive immune cell like a T cell. And that T cell now holds the memory for a long term. So how does this work? For example, if we are infected with a pathogen or an antigen, then antigen presenting cell like a dendritic cell or a macrophage or a B cell that we know from our classical immunology textbooks will take up this antigen. There will be phagolysosomal fusion where there is acidification inside the cell. And this particular antigen is broken down into smaller peptides. These peptides then get loaded on host-derived molecules called MHC class 2. And this gets then loaded on the surface of the antigen-presenting cell for presentation to the CD4 T cell, hence the term antigen presentation. Now, immune system has multiple checkpoints to make sure that we don't have a large amount of response or insufficient immune response. So just the signal 1 from the antigen presenting cell is not enough. So there is a signal 2, which also plays an important role. So there are two flavors of this signal 2. Signal 2 can be a positive signal, which can be CD4 T, 80, 86, or ICOS, which essentially tells the T cell, okay, now you can get activated, you can proliferate, you can produce cytokines, you can differentiate to become a particular subset of T cell and hold that memory. And that is more of an activation signal. Or on the flip side, there is an inhibition signal, which can be molecules like PDL1 or PDL2, which tells the T cells, okay, you should not be responding to this antigen. You have to stop. You have to go kill yourself or become irresponsive for the rest of your life. And that is called as T cell inhibition. And some of you might be familiar with the molecule PDL1 because our cancer cells, when they are forming tumors, actually express a lot of PDL1. As a result of which our T cells, even if they want to kill the cancer cells, cannot, because this PDL1 signal is telling the T cells to get shut down. So clearly you can see that these two signals play a very important role in telling the T cells whether to get activated or whether to get shut down. And once these two signals are delivered, there is actually a signal 3, which is the type of cytokine this antigen presenting cell has to give to the T cell. So depending on the type of pathogen that you're seeing, so for example, if you're infected with an intracellular bacteria like Listeria or a virus like SARS-CoV-2, the antigen presenting cell in addition to signal 1 and signal 2 also produces the cytokine IL-12, and that tells the T cell to upregulate the transcription factor Tbet. And now the T cell becomes a Th1 T cell, which produces interferon gamma, and this interferon gamma potently kills infected cells. On the other side, if you have some sort of an allergic response, then the T cell is instructed to produce GATA3, and now you have all the mucus production and all the bronchoconstriction that makes it difficult to breathe. In context of our biology, in case of an extracellular bacteria like Streptococcus pneumoniae, the T cell produces ROR gamma T and produces IL-17, as you have already seen based on my previous slides. And then there are subsets of T cells that play an important role in regulating these responses. And these T cells express a lot of FOXP3. One thing that I really want you to remember is that our classical immunology textbooks right now tell us that a T cell is programmed to produce only one type of transcription factor and hence can function only in one particular differentiation. So a T cell can only be a Th1 or a 2 or a 17 or a Treg at one particular point of time. And as you will see in the slides following, I think one of our work is one of the early few ones to change that dogma. So we started with a very simple question. We know epithelial cells have MHC class 2 in the lung. can we show that in our hands? Because we have to see if the previously published data is reproducible in our hands. And the moral of the story is using a mouse that can express GFP specifically in the alveolar type 2 cells, we could indeed see that the epithelial cells in context of an infection can upregulate MHC class 2 across all the subsets of epithelial cells, may it be the airway club cells or the ciliated cells or the alveolar type 1 cells and the alveolar type 2 cells. An important side finding is that we also discovered a new subset of lung epithelial cells that expresses high amount of MHC class 2. But given the constraints of the time on this talk, I won't be talking much about that new discovery and the biology relevant to that cell type. So you could have all the MHC on the planet, but does that mean that these cells are actually activating the T cells? And to prove that or ask that question, what we did was we created a mouse where we could delete MHC class 2 specifically in the lung epithelial cells. Once we deleted the MHC class 2 in the lung epithelial cells, we isolated the epithelial cells in a single cell suspension and then cultured the different subsets of epithelial cells with the T cells in a plate in presence of its antigen, and then perform flow cytometry to see if the T cells are activated or not activated in presence or absence of epithelial MHC class 2. And consistently, we saw that deletion of MHC class 2, which is signified by the delta epi, shows that deletion of MHC class 2 consistently prevented activation of T cells across all the different epithelial subsets, suggesting that for the first time, we might have to revisit our textbooks and suggest that epithelial cells can also be professional antigen presenting cells. So now we can have MHC and activate the T cell, but what type of co-stimulatory molecules are these epithelial cells expressing? And we had no clue about this. So we essentially did a time course study where we first took epithelial cells at all the different time points and then used flow cytometry to look at the co-stimulatory molecules or the co-inhibitory molecules I mentioned to you previously. What we did was we took all the epithelial cell type data, put it together in a computational analysis and asked the computer to tell us what type of epithelial expressed what type of molecule. So that way we are not biasing our data by how we want to get our data on a flow cytometric software. And turns out that the software actually could differentiate 7 different subsets of epithelial cells based on the flow cytometry panel we used. One thing that we saw that was really interesting was we saw that epithelial cells in different parts of the lungs actually respond very differently to an infection. So what we saw was the airway epithelial cells. Here shown in yellow is the airway club cells, which are the ones that secrete the vesicles. And the airway ciliated cells, here shown in red, are the ones that have those cilia that constantly beat. These epithelial cells produce a lot of the co-stimulatory molecules, CD4 T and CD86. Whereas the alveolar epithelial cells, which are down there in the alveoli where we are actually breathing, express a lot of the co-inhibitory molecules, PDL1. So this again goes to argue that all epithelial cells are not the same. Every epithelial cell will respond differently depending on the microenvironment that they are in. And what is really interesting is that it seems that the epithelial cells can tell the T cells whether to get activated or inhibited depending on which part of the lung they are situated in. And this was really interesting to us because for multiple decades now, all immunologists know that if you look for CD4 T cells in a lung using CD3 antibody, all the T cells usually are found around the main airways. If you look at the alveoli where there is this high expression of PDL1, there is not a lot of T cells. And we know that PDL1 actually shuts down T cells. And we know that the alveoli is where we don't want inflammation because these are these really thin epithelial surfaces where air actually passes through. So if there is inflammation there, there is a high risk that we might get pneumonia. So our data seem to suggest that epithelial cells are telling the immune cells where to go and where to sit in a lung. So to see if this is really the case, we took these mice where we had knocked out epithelial MHC class 2, hence they cannot tell the T cell what to do. And then we did immunofluorescence to look for CD4 T cells in the lungs. And consistently, we saw that in mice that had MHC class 2, there were a large amount of T cells around the airways and along the airways. However, once we delete MHC class 2, the number of T cells actually were reduced around the airways, suggesting that epithelial cells play a very important role in telling the T cells where to go in the lung and how to protect the lung. So this is really interesting, but then this raises a whole new question, right? Is we have T cells that are sitting around in our lungs all the time, which are presumably protecting us every time we breathe a pathogen. But then this immune response is not killing us every time we are breathing in this pathogen that is very common all around us. So there might be something that epithelial cells are constantly doing to the lung or the T cells to protect us from all that infection related inflammation. So my next question was, if we are inhaling 11,000 liters of air on a daily basis, and this air has cognate antigens that would activate the T cell, is lung epithelial cell playing a role in telling the T cells what to do? We know that the epithelial cell are telling the T cells where to go, but we still are not sure whether the epithelial cells are telling the T cells what to do. So to ask this question, what we did was we took the mice that had or did not have MHC class 2, infected them to induce the T cells in these lungs. So now these T cells are the equivalent of young adult lungs like our lungs are. And then I challenge them with yet other non-lethal strains. So for example, we have these T cells in the lung, we go out in the world and we breathe this pathogen, and this pathogen is not killing us, but deep down our immune response has been activated. So this is what we are mimicking here. And then once the lung was rested, now we look at the T cell with the idea that we want to look at the transcription factors that I had previously mentioned to see if the T cells have been instructed to have a different differentiation program. And again, not to bias the data to manual analysis, we put together all this T cell data and then perform computational analysis. And what we found was very surprising. Within the same lung, we could identify 21 different subsets of T cells based on all these markers that they were activating on their surface. But more interesting than that was the fact that we found that there were T cells like, for example, cluster 4, which were expressing the transcription factor for Th1, Th2, and Th17 biology simultaneously. So this T cell here, which falls under the cluster 4, is a Th1, Th2, and a Th17 T cell at the same time. And this essentially breaks all the data that is currently out in our textbooks, which tell us that the T cells can be only of one particular program at one particular time. What our data is suggesting that in organs where the T cells were not previously studied, like the lung or the skin or the gut, the T cells are actually way more complex than they are in the blood. And that is really indeed the case, because if we look at the blood of these mice, we don't see that level of multipotency or plasticity in those T cells, suggesting that within the same host, T cells to the same antigen are behaving very differently in the lung versus the blood. And that has a large amount of implication for vaccine biology. What is further more interesting is if you look at this cluster 4, which is multipotent, or this cluster 1, which is multipotent, the mice that did not have MHC class 2 actually have higher frequencies of these plastic T cells. So what this is telling us is that the epithelial antigen presentation is somehow reducing the number of plastic T cells in these lungs. And if we delete the epithelial MHC class 2, suddenly these T cells are becoming very plastic and are capable of causing a lot of inflammation. And you can see the confirmation of this using mathematical modeling, where we do see that the deletion of MHC class 2 shown here in red actually leads to an expansion in the multipotency. So this is really cool. So my data so far suggests that T cells need epithelial cells to tell them where to go. T cells need epithelial cells to tell them what to do. But then comes the big question is, how is the epithelial MHC class 2 telling the T cell all these controlling functions? And we stumbled upon this data serendipitously when we looked at the expression of this co-inhibitory molecule PDL1, which I had mentioned you in the past, which shuts down the T cell and constrains the activity of the T cell. What we found that despite whatever epithelial cell type we looked at, we saw that if the epithelial cell increases expression of MHC class 2, expression of PDL1 also increases, suggesting that MHC class 2 and PDL1 always go up or go down together. This was not the case only in mouse lung epithelial cells. We could also take human lung epithelial cells from human donors and show the exact same thing. If the epithelial cells are high MHC, they have high PDL1, so that they are perfectly correlated with each other. And this line essentially signifies that these two epithelial cells came from that exact same patient, suggesting that MHC and PDL1 go hand in hand in case of lung epithelial biology. And what is further more interesting is if we looked at these mice where we had deleted MHC class 2, so we had essentially knocked out the gene for MHC class 2 in the lung epithelial cell, expression of PDL1 on the surface was lost. So I'll repeat this again. We deleted the gene for MHC class 2, the gene for PDL1 was completely untouched, but the epithelial cells that do not have MHC shown here in red actually reduce their expression of PDL1 on the surface, suggesting that PDL1 needs MHC to get to the surface. And this is really interesting. So then the obvious question is MHC class 2 affecting the transcription of PDL1 if it is affecting levels of PDL1? And what we saw was, despite whichever epithelial cell type we looked at and whether the cell expressed or did not have MHC class 2, transcription of PDL1 completely was unaffected. Instead, what was happening was that if we take the mouse lung epithelial cells, stain them for PDL1 on the surface versus PDL1 inside the cell, so PDL1 stained on the surface, so the extracellular PDL1 versus PDL1 inside the epithelial cell, intracellular PDL1, we see that the wild type mouse, so that mouse that does have MHC class 2, can transport up to 75% of its PDL1 to the surface, so that PDL1 can now talk to the T cell and shut down the T cell. However, if the MHC class 2 is deleted, there is PDL1 being expressed. 95% of this PDL1, however, stays stuck inside the cell and never reaches the surface. So if you remember that initial schematic where I showed where the MHC class 2 goes from the rough endoplasmic reticulum to the trans-Golgi apparatus to the vacuoles to reach the surface, it is possible that PDL1 is actually hitchhiking with the MHC class 2 to reach the surface. And if you knock out MHC class 2, now the PDL1 cannot reach the surface, as a result of which the PDL1 probably cannot tell the T cell what to do. So if this is really the case, then we know that PDL1 actually binds a molecule called PD1 on the T cell. So if this is really the case, I should be able to delete PD1 on the T cell and pretty much show all the biology I have shown you so far by leaving the epithelial cells completely untouched. And that was indeed the case. If we took a mouse that is PD1 knockout, so that now they cannot receive the instructions from the epithelial cells, and put them through the same protocol, you will see that the blood of these mice were completely unaffected, so they did not have multipotency. The lungs had more multipotency, suggesting this PD1 actually talks to the PDL1 on the epithelial cell, which requires MHC class 2 to get to the surface. And this was a really cool finding, suggesting that actually epithelial cells, while they are responding to the T cells, are probably more important in telling the T cells what to do by antigen presentation. This antigen presentation not only tells the T cells where to sit based on the level of the T cell stimulation that the epithelial cells offer, but these antigen presentation also puts this selective bottleneck on the T cell, which tells the T cell you cannot become more plastic than this. However, if we are deleting the MHC class 2, the ability of the MHC to take the PDL1 to the surface is affected, as a result of which the PDL1 cannot talk to the T cell via PD1. And now the T cell is not constrained, and that allows the T cell to become very plastic and respond very weirdly. And we think that that is what is responsible for all this inflammation we see in these mice that do not have PDL1. And this is a really interesting finding because in the clinic, anti-PDL1 or anti-PD1 therapy is given to patients that have cancer. And this therapy has shown a lot of benefits to the patient because it really tells the T cells you can go and get activated, the PDL1 cannot shut you down anymore, as a result of which the T cell can kill the tumor and protect the host. But the biggest side effect of this immunotherapy is that these patients have a high rate of what is called as checkpoint inhibitor pneumonitis. And what this means, pneumonitis means inflammation of the lungs, and it is a sterile inflammation of the lung where there is no pathogen discovered. So what we think is happening is when these patients are getting this antibody, while the tumor's ability to shut down the T cell is lost, the epithelial cells can now not tell the T cell not to get activated, as a result of which T cells that are responsible for protecting you in context of allergy and infection see their antigen and become overtly activated, and now the epithelial cells cannot shut them down. So hopefully with these two bi-pronged approach to my talk, I have convinced you that the epithelial cells can tell the T cells what to do, and in return the T cells can tell the epithelial cells what to do, and really signifies that the epithelial T cell interaction plays a very, very, very important role in regulating the pulmonary outcomes to respiratory infections. Now you might think that all this biology is relevant to the lung. How does this apply to all the other tissues? So since our finding, what we have seen is there have been really high profile papers that have come out in really big journals showing that epithelial cells in the intestine, epithelial cells in the skin, and now recently there is a paper out there showing that epithelial cells in the nose, all of them can express MHC class 2, which means that all these sites that are constantly bombarded by pathogens have the ability to talk to T cells. So what we think is happening is a lot of the biology that we ended up discovering in the lungs is probably applicable to all these different organs. And we hope to be able to keep making such discoveries that should be broadly applicable to mucosal immune responses and hopefully be translated to vaccine strategies where we not only target the T cells for vaccines, but one of my major goals is now to try to focus on stimulating the T cells the right way so that they can help the vaccine activated T cells to take home in that epithelial tissue to respond better in the future infections. So with that, I'd like to acknowledge the huge village of people that was very crucial to help me do this job of making these discoveries. All the people that supported me over the years back in my bachelor's, master's, my grad school, and my postdoc, my funding sources from NIH and NHLBI, my current host institution. And with that, I'd like to take any questions, if any. Thank you.
Thanks a lot, sir. I, Altaf Parande, Assistant Professor, Biotechnology Department, would like to acknowledge your presence in today's RIST conference. I'm sure everyone has great knowledge to take away. Despite of your busy schedule and time differences, you made your availability and joined us late midnight. Thanks a lot, sir, for your time and your cooperation that made this session successful. Thank you. We now would go for a lunch break and then rejoin at 2:30 p.m. Till then, stay tuned and rejoin.
Thank you so much. Bye.